will be very interesting, I think. This is one of my first books published in 1999. What we call developed a new concept called as a perinatal strategy to prevent adult diseases. That means whatever diseases we are developing as an adult are predetermined when you are at fetal stage. That means there is genetic imprinting of the adult diseases in the fetal stage itself. So that is why maternal health, placental health, and the environment in the uterus are extremely important, because the fetus grows mostly importantly between the second trimester of pregnancy and the first 5 years of age. After that, there is no further growth in the brain and other organs. They are only gaining the learning, the skills that are necessary for future life. And there is a lot of evidence for this now. So that is why nowadays we give a lot of importance, at least in US and Europe, to the maternal health and the health of the pregnant ladies, and during lactating period, and during the first 5 years of the life of the newborn babies. Then of course we did a lot of work on what is called as metabolic syndrome, which is very common in India. Almost 20 to 30% of the population have metabolic syndrome. That is a constellation of diseases: obesity, diabetes type 2, hypertension, coronary artery disease, and so on and so forth. And it is very easy to detect who has the metabolic syndrome. I can straight away say that in this audience, especially those who are above 40 years of age, 50% have metabolic syndrome. If you look at somebody, and if his abdominal girth is more than the chest, that means he has abdominal syndrome, metabolic syndrome. They have insulin resistance, and if not immediately, they will develop shortly type 2 diabetes, hypertension, and so on and so forth. So that's why whenever I'm traveling, especially with my wife, I point out to my wife, look at the number of people in the airport, how many people have metabolic syndrome. Okay, so by looking at a person, you can make a diagnosis. Then of course we developed a new concept as to what is the molecular basis of health and disease. Why somebody is healthy and why somebody is diseased. What is the difference between health and disease? Various diseases. Disease pertains to all types of diseases, including cancer, metabolic syndrome, type 2 diabetes, hypertension, coronary disease. So we developed markers to say that if these markers are elevated or if some markers are low, then their chances of becoming sick are high. And if some markers are in the normal limits, that means they are healthy. So we use these markers to predict what type of diseases these people have or they are going to develop, and how they are going to respond to treatment. So this is very important. So it is both predictability, dependability, and also reversibility of the disease is predicted based on these markers. And I'm going to talk about these markers shortly during my lecture. Not all, but only at least some. Then of course we recently published another book. I think this is about 2 years back. What is the molecular biochemical basis of cancer? Why a normal cell becomes a cancer cell? Can we convert a cancer cell into a normal cell? Or can we kill only the cancer cell without doing anything to the normal cell? And so based on this, we developed new treatments, and now they are in clinical trials. And we also have the FDA discussions going on now for the approval of some of these drugs. And then of course, another aspect in which we are interested is autism, a mental disorder that is very common in many children. At least 6 to 7 years of age, this can be detected very easily. And at least in USA, the incidence is around 10%. And I'm sure in India, it is more than 20%. In many children have autism, but it is not detected unless they have very gross mental retardation. So if they have any behavioral abnormalities, difficulty in learning, developing skills, they have autism. So they have to be very carefully screened, diagnosed, and then properly treated. So autism is a very important disease because the future of any nation depends on their children, or the young population present in the country. So autism detection is very important. This is our first book on autism, which we published with another co-editor. And then of course now we published a second book on autism last year, and a third book is going to be published next year. So we are giving a lot of importance to autism, because autism is one disorder where there are biochemical abnormalities, neurological abnormalities, and neurochemical abnormalities, and genetic abnormalities. And all these abnormalities sometimes are dependent on the environmental factors, what we call as epigenetics. Not totally genetics, but epigenetics. Epigenetics means genetics are genes that are regulated by the environmental factors. That includes pollution, atmospheric pollutants, and so on and so forth. So because of this interest, we slowly embarked on to look at the other neurological disorders like Alzheimer's disease, how memory is formed, whether memory can be enhanced or not, if so, what should be done, and so on and so forth. So I show you a couple of slides on how to regulate memory, how memory can be improved, and so on and so forth. So we are now coming out with a preparation for this, which we are already giving to some children. Then this is our second book on autism, which we published I think last year. Then of course this is my wife insisted that I show this slide to you. This is the Shantiswarup Bhatnagar Prize, which I got in 1992 when I was a professor of medicine at Nizam Institute of Medical Sciences in Hyderabad for our work on cancer. Then whatever slides I'm going to show you is based on our work. It is not based on hearsay, or it is not based on somebody's work. And most of the slides which I am going to show you are derived from these two articles which are published very recently, called as Biomolecules. I think one is published about 3 or 4 months back, and another was published only last year, where we developed a new concept saying that it is not the DNA or genes that is important for the cell. It is the cell membrane which is most crucial for the cell, to keep you healthy or for somebody to become sick. That is because the cell wall is the one that determines the response of the nucleus or the genes to the external environment, and the response that is depicted by the genes to this external environment. Because all messages have gone into the cell through the membrane, all responses should come out from the nucleus or the genes through the membrane. So it is a membrane structure that is very, very critical. So we determine the membrane structure in several diseases and shown that there have been some very specific biochemical abnormalities in the form of lipid abnormalities in the cell membrane, and this depends on the cell, on the type of disease. It is very peculiar in cases of diabetes, very different in case of hypertension, very different in case of cancer, and so on and so forth. So by looking at the cell membrane composition, we can say that what disease that cell is going to have or going to develop, or what the person is going to have. So we can use cell membrane properties as a sort of a predictor, as a marker to know what is the disease and what is the response of the cell to various drugs we give or the stimuli we give and whatever we are doing to the patient. So this is a new concept which we developed called as cell membrane theory. And of course this also applies to old age. We concentrated mainly on old age because during the aging process the cell membrane becomes more and more stiff. And as a result, the number of insulin receptors on the membrane are decreased, and so the insulin is not able to bind to the membrane properly, and so the insulin message is not getting into the cell properly, and so somebody develops hyperglycemia, and ultimately they develop type 2 diabetes. So if we can modify the cell membrane and make it normal, then this insulin resistance, insulin number of receptors, and then the hyperglycemia, all these things will come out. So we are now trying to work more on these bioactive lipids to see how cell membrane structure can be modified and made into normal, so that several diseases can be prevented and also treated. And this is one of my slides where we got the first prize in the European competition for having developed a new drug molecule for diabetic retinopathy. So this was a worldwide competition which we got it about I think 4 or 5 years back. And then there is another site if you go to Google, called as Innocent. It's a site called as Innocent, where all problems, both in biological sciences, physical sciences, and chemical sciences are displayed. So anybody can compete that throughout the world. So if you have a solution for a problem which they have shown on the site, you can present your solution and say how that solution can be tested and so on and so forth. And if your solution is selected, then they'll give you, money in dollars. Okay, so we developed a new marker for diabetic nephropathy, and that extended to the renal failure. And so we won the award, and we got some money. I don't know how much, but we spent away the money anyway. So all this work is not possible without collaborations. So we do a lot of international collaborations. This is one of my groups working in Hungary, a group of about 25 to 30 scientists working with us on cancer to develop new treatment for cancer. So here you see that most of these scientists are not even biologists. Some of them are computer scientists, Some of them are mathematicians, some are the statisticians and physics persons, and so on and so forth. So if science has to advance, the collaboration should be across the sciences, not just biological sciences and only in the life sciences and so on and so forth. So we collaborate with a lot of scientists who are mathematicians, statisticians, and computer scientists, and try to develop solutions to the biological problems. Then this is my group in China with whom we collaborate, Professor Shen, who is standing next to me, and his PhD students and postdoctoral students. So there we work mainly on diabetes and diabetic retinopathy. So this is a university called as Zhejiang University. This is the second world's oldest university. It's more than 400 to 500 years old. And if you go and visit that university, it is like a city. It is a very, very huge, and each department has one separate 4 or 6 stories complex. And they have their own 5-star hotel so that any guest coming to the university can stay in that hotel. So when they asked me to stay in that hotel, I asked who is running this hotel because it's so good. He said it is our university hotel which we run for the sake of our visitors. Then it is an international group with whom we are collaborating from Argentina. So Professor Aldo Aynad is my collaborator, and there we work mainly on bioactive lipids. So measuring them, titrating them, characterizing them, identifying their structure, and so on and so forth, and his enthusiastic PhD students and postdoctoral students. And you find that in most of these slides, the students are mostly females. Everywhere in the world, one phenomenon that we are observing is there are less and less number of male academicians and more of female academicians. That is what occurring everywhere, especially in USA and also in India. Then of course, I showed you those international collaborative slides because the diversity is very important. People coming from different backgrounds, different countries, and different cultures and backgrounds is extremely important for the exchange of knowledge, developing new ideas, and developing new concepts, and developing new treatments for any disease. And this is what that is at. And in fact, in Canada, when I was working a long time back, our lab used to contain only 6 scientists, and each scientist from one country. I was from India. At that time, I went to Canada from India. And then we used to have scientists from Australia, one from China, and one from Mexico, and one from Chile, and one from Middle East, a Muslim scientist. So the institute believed that there should not be more than one scientist from one country. So all countries are represented in the institute. So this diversity and exchange of information from diverse background people is extremely important. This slide is not okay. So now let us see what happens from health to disease and to death. So you do not become sick in one day or in few hours, except when there are acute infections. Most of the time, so-called lifestyle diseases which somebody talked about in the morning, like metabolic syndrome, insulin resistance, type 2 diabetes, hypertension, cancer, all these things, they start very, very early in your life. When you are 20 or 30 years, you would already develop insulin resistance. But you become a diabetic only when you are 40 or 50. That means it takes about 10 to 15 or even 20 years for you to become a diabetic. So just because your blood sugar is high now doesn't mean that you developed yesterday. That means it is going on for more than 20 years, sometimes 15, 20 years, and so on and so forth. So nobody is sick immediately, except when there is infection. So cancer also same thing. The cancer cells are already present in your body, and then they grow, and they have to attain a specific size, and then only we are able to detect by MRI and CT scans. And that means by the time the cancer is detected in an individual, that means he already had the cancer for the last 10 to 15 years. Only thing is now we are able to detect because he got the symptoms, signs, and then he came to the hospital for review and so on and so forth. So nobody is sick suddenly. And so health to disease takes from hours to months to years. So if we can understand the biochemical pathway, what are the molecules that are involved, what are the genetic changes that occur in the cell that convert our healthy cell into a tumor cell or a cancer cell or a diseased cell, we can take the same path and then just reverse it from disease to health. It is possible. Nowadays we have the technology to do that and all those things. I will show you some slides how we can do that. And so in the same manner, the recovery from the disease to health also takes time. It is not overnight. So when our patients tell me that, sir, I have been in the hospital for 1 week, when I'm going to become all right? So I have to tell him, you have been sick for more than 20 years, and so you cannot ask us to make you all right in 1 week. So you have to explain to the patient what is the sequence of events that occur and that make you sick, and then so how easy or how difficult it is to recover from sickness to become healthy. So let us say, take the example of inflammation or injury and our body's response. So whenever you have an injury, then injuring agent, we call it as inciting agent, it produces an injury. So if you have a accident, for example, so you fall on the ground and then you have an injury. The injury may be because you have fallen on the road, you have fallen on the motorcycle, or the car has fallen on you. So it produces an injury. This injury leads to reaction on the part of the body in the form of what is called as inflammation. That means the tissues are literally inflamed. That is why it is called as inflammation. Inflame. So the tissues are burning. They are secreting molecules which produce damage to the tissues which are injured. At the same time, molecules that are necessary for the resolution or the healing of the wound also are produced simultaneously. And in this process, for the healing to occur, the removal of the inciting agent is necessary. That means you have to get out from the accident. And then all the injured tissues should be removed. And that is what is called as resolution of process. So that means inflammation should resolve, and so that the inflamed tissues will come to normalcy. And then that is not sufficient. Then what is called as the healing should occur. And for the healing to occur, there should be regeneration of the tissues. Either there could be regeneration of the normal tissues, or sometimes the normal tissues are destroyed so much that they can be replaced only by fibrous tissue, so you develop a scar. So people think that when you have a thorn or a injury to your skin or to your body, if you just remove it, healing automatically occurs. It doesn't. You are only removing the inciting agent, so the inflammation will continue. And so the removing the inciting agent is not sufficient. Then there should be resolution, for which separate molecules have to be produced by the body. So they are called as the molecules involved for resolution of inflammation. And then healing occurs, so the regeneration tissues should occur. And then, of course, once the regeneration occurs, the regenerated tissues should get integrated into the surrounding normal tissue, so that homeostasis is established. So it is not sufficient, if just the regeneration of the tissues occur. They should get integrated into the existing normal tissue, surrounding normal tissue, and so there should be nerve supply, blood supply, everything, and should get into the normal routine of the body. And that is what is called as the restoration of the homeostasis. This should occur. And this is most evident whenever you have a renal transplantation, for example. You remove the old damaged kidney, you put a new kidney. But just putting the kidney does not make it function. That kidney should get enough amount of blood supply. It should get the nerve regeneration should occur, and the blood supply should go to the kidney, and the kidney should accept it, filter it, and then get into its normal function. So if today transplantation is done, he will not pass the urine next day. It will take about a week or so. Meanwhile, the body tries to recognize it as foreign and try to destroy it. So that has to be suppressed. So you have to use immunosuppressive drugs. And so the establishing of this homeostasis takes time. So in this sequence of events, every stage is controlled by a set of molecules, genes, and so on and so forth. And so if you can understand these molecules, the pathways involved in this production of these molecules, what are the overlapping features between inflammation and resolution and homeostasis, and what are the cellular structures that are involved in this process, then we will be able to mimic it. So you mimic the nature in which the healing occurs, and then try to follow it and try to implement it in the patient so that the patient becomes normal and recovers from the disease. So, but these events occur in such a close interaction fashion that there is cooperation, there is collaboration, and both positive and negative regulatory events occur. And they occur in a very timely fashion, sequential fashion, and in a coordinated fashion. And unless all these events occur in such a fashion, the health cannot be restored. So there is a saying which I was reading some time back. So whenever you see the facts, then you learn. So what you are learning in your school, in your college, or in research is the facts. You are learning the facts, and so you learn them. But if you see the truth, then you start believing it. That means you experiment it and you see for yourself that is truth, and then you believe it. But if you tell this learning process, these facts and this truth in the form of a beautiful story, you will remember it forever. That is why there is difference between teacher to teacher. Some teachers can tell you your subject in a very beautiful fashion, and so you'll never forget it. Some teachers will just tell like a gramophone record, then you will not remember them because they are not beautiful. They are not so nice to hear. So science is about facts. And so we learn science every day because there are facts every day in our life. And when you see those facts and believe them, then you will believe them. So that is why this is very important. So learning, then seeing the facts, and then seeing the truth, and then remembering it in the form of a story so that you will remember forever. Then of course there is always, people talk about ancient wisdom and so on and so forth. So that the question that occurred to me is, can ancient wisdom become old or ageless? Can wisdom ever become old? Like we people becoming old sometimes with age and so on and so forth. Can technology become old? So we call it, oh, that is old technology. So my students, when they look at my cell phone, they say, this is old technology, you should get a new cell phone. But remember that, like, the old technology is necessary to develop new technology. So unless you know the old technology, you cannot develop the new technology. So this is very important. So science and technology are forever evolving. Then of course, you also have the spirituality, or what is called the spirit of science. So we don't call it as spirituality, we call it as spirit of science, or science of spirit. So science and spirit and spirituality, there is no difference if you really understand science. Okay, so that is what is very important. Then of course, this is what I'm showing that how somebody is sitting and polishing his shoes, and instead of doing it with the hands, he is doing it in a machine. So after polishing your shoes with hands, then the machine has come. So polishing the shoes with hand is a old technology, with a machine is a new technology. But hands will never tire, but machine can break down. So I asked my wife, why Ramayana and Bharata are so popular? Why people believe it? She said, oh, they are epics, they are facts, and so on and so forth. Then I said, they are beautiful stories of facts and truth. And that is why you never forget it, and they are told in a very, very beautiful fashion. So your science learning also should be like that. You learn the facts, you learn the truth, and then learn it in a storytelling fashion, then you will never forget it, and that is how the students should be taught science. So that is why I like this saying. The key to the future of the world is finding optimistic stories and letting them to be known. So always have optimism that the science can do this, I can do this, and society can do this, and government can do this. Then there will be future. Then of course, whenever you develop a new technology, it should not pollute the environment. That is very important. Then whenever you are in difficulty, you should not feel bad, but try to overcome the bad situation. Then of course, you should also have happiness. And with happiness will come from science and science of happiness. Then people generally tend to believe that happiness is money and all the physical features you have and so on and so forth. Back in 460 BC, Democritus, on whose name we call it as democracy, he says, happiness resides not in possessions and not in gold. The feeling of happiness dwells in the soul. That is very important. So you should feel happy that whatever you are doing is good, and so on and so forth. And when I talk about this happiness, I always wonder what makes a person happy. Why our neurons become happy when we do something and also. So for a hungry man, if you give food, he becomes happy. That is because the dopamine levels in the brain go up. Dopamine is called as a happy hormone. So and then there is also a disease called as reward deficiency syndrome. Most of the politicians suffer from this. No matter how much reward you give, he is not happy. With 100 crores, 200 crores, 500 crores, they are not happy. So they don't have enough dopamine in their brain. So we should measure the dopamine levels in their plasma and see whether they are happy with what they got or not. So we do these dopamine levels in certain clinical conditions. Like, especially whenever we want to see whether the stimulus that is given to the body is sufficient enough for the resolution process of the wound to occur, and so on and so forth. So putting all these facts together, we want to develop drugs. Put all this knowledge together and develop drugs. And developing drugs is not easy. It is a 4 or 5 stage process. So first you have to discover a molecule. And to discover a molecule, you should understand the disease process, the molecular biology of the disease, and then you should develop what are the new insights into the disease, and then see what are exactly is the wrong things that are happening in this disease process, and try to see how you can intervene or how you can rectify it. Depending upon that, you develop a molecule and see that molecule is working in vitro, and then in animal models, and then human clinical trials. So whenever you want to do clinical trials, you have to do what is called as preclinical testing, what we call as toxicology studies. If you go to FDA website and look for drugs, there is a column called as drugs that are not effective but not toxic, but they are approved. So FDA will approve any drug as long as it is not toxic, even if it is not effective. Remember that. So the first important thing that FDA looks whenever you file an application is whether it is toxic or not. It is not, they are not bothered whether it is effective or not. But if it is non-toxic and at the same time effective, that is good, because then you can catch the market. Then of course, after you get all this data and everything, you prepare what is called as investigational new drug application. That is called as IND. So you prepare a bit brochure of about 1,000 pages or 1,500 pages and go to FDA saying that this is our molecule, and this molecule is developed based on these facts and this truth. This is the molecular pathway it works. These are the preclinical toxicology studies, and these are the animal studies. And the animal studies we have shown the efficacy, non-toxicity, and so on and so forth. Then they will review it, and then they will give you permission to do clinical trials. So whenever you want to do clinical trials, then you have go through 4 or 5 stages: stage 1, stage 2, phase 1, phase 2, phase 3, phase 4. Of course, phase 4 is only a post-marketing surveillance. Phase 3 is the most important, where you do double-blind placebo-controlled trials. And then, of course, during the trial, you have to do a lot of other investigations, like whether the drug is getting absorbed as predicted or not, what are the plasma levels, what is its metabolism, what is its half-life, whether any tissue specificity is there for the drug or not, whether in which tissue it gets mostly concentrated, and if it gets concentrated, how long it is going to stay in the site or not, and so on and so forth. We learned most of these things because we were developing a drug for glioma, brain tumor, and so we went to FDA, and so we had to give them the data about, even though we are injecting into the brain, into the tumor, into directly our drug, the FDA wanted to know whether the drug is going to spill over from the brain into the peripheral circulation. We know that it doesn't come out, but still they said you have to measure it in the plasma and show that there are no changes in the plasma levels of the drug before and after injection into the tumor. So we have done that. Then of course they want to know how much drug is there in the tumor, how much drug is there surrounding the tumor, and what are the metabolites that are formed, whether those metabolites are present in the plasma, and if though they are coming into the urine or not. So no matter where you inject your drug, FDA wants all this data, and you have to produce it and show them. And FDA is also not bothered to some extent if there are side effects, because there is no drug which is not without side effects, depending upon the dose, the duration of treatment that is given, and so on and so forth. As long as you identify the side effects and warn the patient, and you know what to do when the side effects occur. So it may be dose dependent, it may be time dependent, it may be metabolite dependent, and so on and so forth. So you should know what are all those things. So once all this data is done, and then you go to the go to the FDA and say that we filed what is called as new drug application, and then do it. For example, we are now developing certain essential oils as the drugs, and so we are going through all these things. Then, so the advice is that follow the nature and mimic the nature. Nature is nothing but our body. So that is how beta blockers and H2 blockers have been developed. Then I want to talk very briefly about diet and exercise, which people have been talking about it. We know that with the diet restriction, you can prolong the life, even including the human life, by 20 to 30%. That's why one of the advices that is given to most of the patients is you eat less. You cut down your calorie intake by 20 to 30%, so you live longer by 20 to 30%. When you eat food, you generate a lot of free radicals. And these free radicals, when they are excess, they are harmful, whereas when you drink water, there are no free radicals developed by this slide. And whatever food you take, you generate free radicals. So that means food itself is toxic, but we have to eat food, so it has to be eaten within normal limits. For example, we had a patient who had a 350 lbs weight. He came to the hospital saying that I want to lose weight. We told him don't eat any food. So he did not eat any food literally for 1 year. He took only water and vitamins and minerals. And after 1 year, he lost 100 lbs. In all this 1 year, he went to the office and worked. So if you stop eating food for 2-3 days, you are not going to die or anything. But people are afraid to miss breakfast and lunch. Okay. And then, of course, we showed that whenever you do exercise, you suppress production of free radicals, and it is anti-inflammatory in nature. So that is why, in fact, we have shown that whenever you do exercise, you enhance the activity of two enzymes called as desaturases. They lead to the generation of a compound called as lipoxygenase 4, which is a potent anti-inflammatory molecule. So how much exercise you have to do? So we generally recommend people to do 5,000 steps per day, but if you can somebody do up to 30,000 steps per day, that is very good. And then, of course, whenever you do exercise, you improve your memory. So this is an experiment we have done in the animals where we have shown that the sedentary life in the upper borderline, the lower room, when you do exercise, the number of neurons and the synapses are increased, so you have better memory. So we tell nowadays to all our students not to study 1 week before the exams. Do exercise, eat less food, and be relaxed, and you write well in the exams. Then, of course, whenever you do exercise, you generate a lot of molecules called as BDNF, lipoxygenase 4, nitric oxide, and so on and so forth. So we measure these molecules, including cytokines, IL-6, TNF-alpha in the plasma, to know whether the exercise done is adequate or not. So if there is changes in the levels of these molecules, then we know that the exercise that is done by the person is adequate. If they are not up to the mark, then we tell them that you have to do more exercise so that the molecular levels are increased. Then, of course, we have developed two molecules called as DH and EAA, which can improve memory. And so we are now making it into capsules, and then we are trying. So what are the 3 advances that have occurred in the last 1 month? So you can develop, people have developed what is called as a biodome. This contains 5 molecules, and you take a frog, cut off its limb, and then put this biodome on the limb that is left after cutting. And in 1 year, the limb regrows completely, including bone, blood vessels, nervous system, everything. So you can see that actually there. All right, the last 4th one. Okay, so that means now we have the technology to regenerate tissues, including limbs, fingers, liver, kidney, especially liver, and so on and so forth. We are trying it for a heart. So that means you can probably use the same technology and same molecules to regenerate brain also to limited extent. Then this is the slide showing that the limb has regenerated and became completely normal. Then, of course, an adhesive type of ultrasound is being developed, which contains 3 or 4 layers of adhesion tissue. You can put it in the stomach, and it continuously monitors the internal organs. And then you can see what is happening to your heart, your liver, spleen, or splanchnic circulation, or alimentary canal, whatever it is. And you can put it anywhere in the body. It is just like a thin layer of film. You can put it, so ultrasound, so if in future we don't have ultrasound machines, you can put this layer, and then it will take the ultrasound of all the features. Then, of course, another thing is how to improve memory. The most important structure in the body that has memory is DNA. So now computer scientists are trying to develop DNA as a memory chip. So you take a synthetic DNA, inject into the cell, it will go and integrate it into the cell DNA, and then it will continuously monitor the cell functions, and then it memorizes it, keeps it in memory, and you can retract that memory and use it when the cell becomes sick. Because by looking at that DNA memory, you know where and when it has gone and it has changed. And so you can go back to that situation and then repair the process, and so the memory can be reborn. So this can be used for the cell memory, it can be used for tissue memory, for organ memory, and for brain memory. Thank you very much.
Any question from the audience?
Of course, I have skipped some slides, but you should pardon me for that.
Thank you so much, sir, for igniting our mental horizons. I realize is a true scientist spirit in you. I wish your wisdomful words and the talks will always be available with us. Thank you so much, sir. एक बार जोरदार करतल ध्वनि से स्वागत कीजिए. धन्यवाद ज्ञापित कीजिए डॉक्टर UN दास जी का, जिन्होंने एक इतने अच्छे नोवेल कॉन्सेप्ट पे हमें जानकारी दी. कार्यक्रम की श्रृंखला को आगे बढ़ाते हुए मैं मंच पर आमंत्रित करती हूं प्रोफेसर राखी चतुर्वेदी जी का. शी इज द प्रोफेसर एंड हेड एट द डिपार्टमेंट ऑफ बायोसाइंसेज एंड बायोइंजीनियरिंग IIT गुवाहाटी. She is a visiting professor in Gifu University, Japan, and is a recipient of Professor F.C. Stewart Memorial Award 2021. Along with that, she is a member of National Academy of Sciences, as well as Plant Tissue Culture Association, and many more. I welcome you, ma'am, and I'm sure that your wisdomful words will be very beneficial to our budding scientists. Welcome.
Yeah, thank you very much. Yeah, thank you very much. And first of all, I wish to thank the organizers and the Patanjali entire team who have invited me to share my views and ideas and then my research. Particularly, I wish to thank Acharya ji and Swami Ramdev ji who have, I mean, entering into the Patanjali itself is a very positive aura I felt. I also wish very happy birthday and wish you very, very happy long life and very happy, healthy long life to Acharya ji, whose birthday is coming. So with this, I think let me come back to my topic here, which I wanted to share with you. There are the two things here. You may see this plant tissue culture and metabolite production. Then we are talking about the sustainability. So sustainability of metabolite production. Metabolite means what we are using for, since for the last 2 days we are talking about the Ayurvedic medicines, and we know all is what we are talking about, the plant-based. And think about if we continuously using these plants from the field, what will happen to the plant vegetation? It may go on extinction, it may, it may deplete. So there has to be an alternative solution. When we are using something, there has to be a replacement as well. So here the tissue culture technique has come up as a rescue to come up as a compensation of the plants which we are using, and side by side as an alternative method to provide the raw material for large scale, commercial scale production of the metabolites. And that's what about the sustainability. This is the first, the tissue culture concept came. See, when we are getting, because a lot of students are here, and I wish to address more to the students because this is something fascinating. And most importantly, when you are using the technology, one should know the know-how of what is what, the basics. And the plant system, you have, most of you have been taught the botany, which is talking about the classical botany. So when you are implementing the technology, one should know about each and every part of the plant system, then the technology work well. Suppose if your vehicle got damaged or punctured, if you don't know what to fix it, what the role of which part, it's very difficult to fix it. So the same is applying to the plant system, and when you are confused with the plant system, just think about yourself, because both are the eukaryotic system. They feel, they sense, and the more they are taking care of it, they will flourish. So based on this, the first concept of this totipotency has come up from Dr. Frederick Stewart, and it was an accidental discovery when he has cut the sections of the carrot, gajar, sections of the carrot in a nutrient medium, and then he found the cells are dividing and they give rise to the embryo. Embryo is a seed. In a layman terms, we call it as the kernel, which is present inside the hard seed coat. जैसे आम की गुठली के अंदर जो soft part होता है, kernel कहलाता है. So we get the embryo without the hard coat in the tissue culture system. So this concept came from the cells, that cells are totipotent. That means each and every cell of the plant system has the capacity to regenerate the lost organ or the entire plant system. So that's the beauty of the entire plant system. So with this concept, you think about, you see here, the lot of techniques are available. But again, this is lot of, when the lot many options are available is equal to no options, because confusion starts. So what I hear is, alert is, this technology is available, but where has to be implemented, what actually is needed in the plant system is very, very important. Because right now, the tissue culture in most of the cases is you see the failure majorly because it has been arbitrarily used without understanding what is exactly needed in the plant system. So the moment we know what is required in the plant system, then accordingly, the technique will be picked up. So here I will entire my talk. What we do the research is, we are not using any foreign DNA element. We are using the existing plant machinery of the same plant and using it in the best way to meet the demand or improvement in the plant system. So these are some of the plant system which we use is, this is the first on the top corner, left corner. This is the tea. We are very familiar. Every day we take this beverage. If you see, there are only 3 parents, actual parents of the tea, and one is Camellia assamica, which is grown in the Assam region of the India, in the northeast region. Other is Camellia sinensis, and the third one is, again, the lineage of Assam, Camellia assamica subspecies lesiocalix. But today, there are many species are available, 320, 325 plus, just because it is aggressively crossbreed. And as a result, in the nature, none of the pure breed types are available. So this is the concern. What is impure is adulterants, because it's a cross-pollinating, and some other species, pollen grains, are fertilizing, and the seeds, propagation is by the seeds, and it's all mixed up. So here we work on the tree species. We work on some plants, but these are the representative. Just wanted to tell you all that we work on the tree species, which is really challenging, because in nature, the trees grow slow, and same reflection is there in the in vitro system. So one has to be a little more patient. Grasses grow very fast in the nature, so in the tissue culture system, we get the same response in 1 week for the grasses, but for the trees, it takes 2 months to complete the one single experiment. And many experiments are to be done to come up to the conclusion point. So we work on the tree species. Another is our own plant, Acharya indica. And when we talk about, everybody says we are very much familiar of it. But unfortunate part of it is still, if you buy the azadirachtin, which is the main component of this neem, there's only one species, Acharya indica. And if you see the literature, it has come from Azadarakht. Azadirecta is Azadarakht is the neem. The unfortunate part is, if we are going to purchase the azadirachtin, 500 milligram comes in around 5,000 rupees still. While we say the plant is growing in our courtyard everywhere, but that's not the fact. It doesn't grow in the cold climate. It likes the very warm temperature, 45 plus. If you go to the southern part of India, it flourishes. So here we are working on these 2 tree species to come up with the pure breed lines so that they produce the constant metabolites irrespective of fluctuations in the nature. The other plant is lantana, where in some part of the, particularly in the northeast, people are using as a datun, as a toothbrush, and it heals the cavity. So idea was what compound is present here. The third one is spilanthes, is anti-malarial, and it is commonly used, the paste is used, and in fact, it is also repel the insects. The Tinospora cordifolia, everybody knows about the gilloy, immunity improvement, it favors. Other than this, it has the anti-diabetic compound. So these are some of the plants. So the trees, the shrubs, herbs, and woody climbers. We work on all these aspects, and we divide our work into 3 major, as and when the request comes. One is the quantitative improvement. That means we need the large-scale propagation of identical plants which are identical to the parent. You don't have to recheck, re-cross-verify. It will be the clones of the parent plant. The second is the qualitative, where we have the parent plant, but we need certain improvement. then we go accordingly, and metabolite production. So this is under the qualitative improvement, quantitative improvement. This is the entire plant body is here. The cursor is not working, but you may please try to understand. The top part is the shoot tip, the tip region, which is known for the two concepts. One, generating the identical plants, and also the virus-free plants. Why virus-free plants? Normally, virus is percolating from roots to the up through the conducting strand. It's like a pipeline is taking the water from to your home. So here, in this tip part, the virus is not reaching because vascular tissue is not there. So this particular part, if utilized, it can generate the disease-free, particularly the virus-free plants. And if rest of the plant, nodal segments, if utilized, it can generate exactly the clones of the parent plant. So what is happening in the nature, what is conventionally happened, we are mimicking some of them in the tissue culture with a little faster speed, where lakhs of the plants can be produced. So the nodal segment, if you see the gardener chops the twigs of the plant and it grows into the hedge. झाड़ी की तरह से grow करता है, if अगर हम tip part को cut करते जाते हैं, trimming करते हैं. So the same concept was taken, but in the conventional method, you need the big twigs to, like vegetative cuttings, in rose cuttings, you need 30 centimeter long cutting and give rise to one plant, that too in the favorable season. So here we are irrespective of whatever the season outside, we take the single node, 1 centimeter node cutting and give rise to it gives several plants. Overall propagation system is divided into two. One is the challenge is to get the shoot proliferation. So here the challenge is what explant part of the plant is to be taken. And that will be decided what exactly needed in the plant. The second is the shoot elongation. The moment you have the shoot, it is to be elongated sufficiently so that it can be placed into the rooting medium, so that the roots develop from the base of the shoot. And finally, the hardening, because everything is happening under the controlled condition of 25-degree temperature and 100% humidity, 16-hour light, 8 hours dark. So here, suddenly if we transfer this into the light or the natural condition, the plant will not survive. So it's a gradual process, a week to 2 months. It's simply, if we are sitting in the air-conditioned room here, suddenly if you go in a chill heat, some of them will collapse or started sweating. So it's always a gradual process for the plant as well. So this is the one method. Here the cutting, you see the corner, left corner. This is the explant we use after chopping the leaves, and the shoot is growing from that particular position which is pre-existing in the nature. Only we are improving the efficiency while providing the nutrient medium. And entire shoot is developed, and then the root plant, complete plantlet is ready in the last picture here, and then finally it is hardened. The second, this is the, from the same nodal segment, we have generated the root biomass, because root is another storage organ for the metabolites. So in plant system, there are 3 points of origin where the concentration is very highly present, high amount is present of metabolites, is the roots, the leaves, and the embryos. So it does not mean it doesn't have in rest of the plant body. It is present, but we target the concentrated amount where it is present, and that's a little strategically plucking the part of the plant for metabolites. So this is the root biomass we have generated. This is the stevia, where again we have used the auxiliary shoot, which we were supplying to the farmers every 12,000 plants per week. Our MTech student was doing it just to help them to grow them large-scale propagation, because here the leaves are to be utilized for steviol glycoside production as a raw material. This is the banana, where we are using the shoot tip because banana have a lot of problem with the viruses. So here we are using the shoot tip. Normally, the plants grow through the sucker, and from one shoot tip you can get only one plant conventionally. So here we can see the picture 4, you can get multiple shoots from the single tip portion. This is the leaf part. We made a disc, so it's the designing of the explant. Otherwise, it can be used the chopping of the leaves. And if the right formulation is added into the nutrient medium, they also develop the large number of the shoots. Number can be increased. This is about the embryos, is like a seed without the seed coat, hard seed coat. So they also are like a bipolar. When you sow them, how you get the plant? When you sow the seeds, the complete plantlet is developed. The same here. When you sow them, entire plantlet is developed. So here one cycle is reduced. When you get the shoot, it is to be transferred to the root and then the plantlet. Here, we are directly moving to the plantlet because this embryo is bipolar, so it is little more faster, and it is produced from single cell, so it's the clones of even the cell system. And large scale propagation can be done. The synthetic seed is produced, nothing is synthetic except the coating. Inside what you see, the green color is actually an embryo, is coated with in the gel and for the storage purpose. And when it is sown into the soil, it gives rise to the entire plantlet. But before we do, we ensure that embryo should be germinating. This is the callus, is more resembling like a cancerous growth in the human system, which is, nobody likes it, but in the plant system, it has two-way advantage. If you see the upper part, is the green color, which give rise to the shoots, and this is known for generating the variations because they divide similar fashion like a cancerous cell, uncontrolled mass of cell division. If we add the right formulation, it will give rise to the shoots. The brown color callus, it's a substitute for the raw material like a cell biomass for metabolite production. So the the important point here is, here the small unit of the cells are present, and that will help to get the efficient metabolite production, because when you are adding the solvent, it percolates into each and individual cell and withdraws the metabolite. Otherwise, if you collect the plant material from the nature, it has to be chopped, but even after chopping, one cannot get the fine bunch of the tissues or the cells. This is the qualitative improvement where we have worked on the tree species, because trees, the bigger challenge for the farmers is in the breeding program, they grow very slow. Suppose if you have planted a mango plant, the reproductive phase comes in 3 to 5 years. That means you will get the fruits after 5 years and the flowering as well. So the farmers have to wait for 5 years to get the flowers so that controlled pollination or manipulation can be done. Again has to wait for the seed to be sown, and then in this way, 6 to 7 generation has to be crossed to confirm is what the traits have been wanting, it is permanent, because it is percolating from generation to generation. By the time it will come, it is 30, 35 years, counting that average is 5 years for the reproductive phase. That's too much cumbersome, and by the time we may forget also what we wanted in the plant. So with this concept, the tissue culture is providing help. Here the inside, this is the flower structure, and this is a shoe flower, hibiscus, it's called Gudhel. The anther, inside the anther is a uninucleate pollen grains are present, and if we get the plants from them, they will be N, half of they are gametes, and which are actually meant for fertilization, but we get the plants from them. So similarly, the egg cell also serves the same purpose. The endosperm cell has a 3N nucleus, very nutritious, like coconut milk is a 3N. If we get the plants from them, it will be seedless plant. So this technology has already been implemented in some of the watermelon, the tarbooj, which you find. Where other seeds are obstruction, the endosperms are grown, cut. So this is the method we use for anther culture, where you see the inside numerous pollen grains are there. They are the male gamete, which is meant for the fertilization. But what we do is, when they're very young, we divert we give them the stress in a way, they start producing the plants rather than participating into the fertilization. So here the stage is very, very important. It should be very young stage. It's very easy to change the mind of the toddler than the mature person. So the same we implement the idea. This is the embryos. Large scale propagation happens when we use the pollen grain in case of tea, and they mimic the same stages like a natural embryo, from globular, globular heart, torpedo, and then the dicot stage. They germinate like a natural embryo. Right now they are haploid. We made them, treat them, this plant, with a colchicine, again a plant-based product, and it doesn't create any abnormality, only it hit the spindle, which is meant for pulling apart the chromosome. So when the spindle is broken, the chromosome does not move apart, they get separated, chromatids, but remain in the same cell and it get doubled. So it's called doubled haploid, so that they normally participate in a fertilization process and bears the seeds. Otherwise, haploid plant will also grow very normally like a parent plant, but they will not bear the seeds. So if we want a seed, it has to be doubled. So this is the neem nemoli. If you see, there are only one species, but still a lot of variation. Why? The same happens with the mango also. Why? Because it is cross-pollinating and unknown pollen decide the quality of the seeds. So suppose if we are interested in, this is the parent plant, and this is the bigger size of the nemoli, and we want some clones of it, we can implement the technology. If we want the pure breed types, we can target the pollen grains or the egg cell. Why we are targeting the nemoli talking about? Because embryo inside is known for all medicinal properties, maximum. In fact, it is used as a biodegradable biopesticide. It degrades on its own in the nature. So this is on the left side is the culture room, and where we are growing after inoculating the explant under the control condition. And once we get the results, and hardening process done in the next right side picture is for the greenhouse, where a large number, once they survive in the greenhouse, they can be transferred to the field condition. So this is normally it is growing in the field, and some changes you see in the lower part, there's 3 leaves are there. The center one is the natural plant, a small leaf, but sideways you see the haploid plant, bigger size of the leaf. So what is happening? Generally, diploid have a larger leaf, haploid have a smaller leaf, and triploid has even larger leaf, but here the reverse is happening. The reason is, in diploid, there is a dominant and recessive chromosomes are present. Dominant is always dominant, and recessive never get expressed. So what happens when we develop the plants from the haploid, both get separately, independently, are happily expressing itself. And probably this larger size of the leaf could be a recessive character, could not get expressed in the cross-pollinating plant. So this is the third which have idea. What we do is, once we have a callus, it can be directly processed as a cell biomass, or further fine suspension can be prepared in a liquid medium where there is no agar, and the rest of the technology is what the chemists follow, is solvent extraction, then lyophilization, or vacuum evaporator, the solvent get evaporated, then TLC is the quick checking, then HPLC, NMR and all, to find out the known or unknown compound. All technology is well standardized, only we have to rightly implement into the plant system. That's what takes time. It's all about the research. So this was the case study, case of lantana. Actually, it is an obnoxious weed in most of the part of the UP, because it's difficult to uproot it. So somewhere in the western UP, some lightweight furnitures are prepared. In the northeast, people are using it for the twigs to heal the tooth cavity. So with that idea, we started working on this plant, and we utilize the flower, the leaf. This is the leaf disc from where we get the callus. This is the stem part, because twigs were used, so we thought something might be there in the stem. The callus is produced, and this is the leaf and the flower. And all of them were producing the compound, and this is the flow chart. So here, these are the target, the leaves, the stem, roots, and the seeds. So this is the callus, because medicinal compounds normally have a lot of phenolics, the browning, and it's a defense mechanism for them. Whenever they get wounded, they first try to heal the wound, like what we do. So in this process, they release the phenolic compounds, but tissue culture they don't like it, because it's an inhibition for fast division of the cells. So first challenge was to remove the browning of this callus. So we started fast multiplying, and at the end we got the very fresh, friable green color texture of the callus, and that's the right texture for processing for metabolites. We followed all this technique of purification and reconformation, cross-conformation, what compounds are present, and some fine-tuning can also be done the moment we know which stage, how many days it takes for fast multiplication of the cells, because the plant cells, they grow slow compared to the bacteria. Because bacteria, they grow in just 4 hours as well. And that's what we have been asked to brush your teeth after having food at night. But for plants, it takes a week or 21 days, 3 weeks. So the moment we know which part of the growth cycle have the fast multiplication, and the fine tuning can be done at this stage while incorporating some bacterial spores or some foreign DNA, or their products, because the moment the fungal spores or their products are added, the cells started taking it as a defense mechanism, and they start producing the metabolites, which are using it as a common for medicinal purpose. Otherwise, they do not pre-exist, all these medicinal metabolites normally. When the plant gets stressed up, senses something attack has been happened, and then it started producing. So thinking about this, we started implementing this. The commercialized scale of production of the cell biomass can done in the reactors 3 liter to, we can grow slowly to 5 liter to 10 and then 100 liter reactors. So with this procedure, we found there were 3 compounds were there, and these are the 3 compounds. They were pentacyclic triterpenoids. They were very difficult to synthesize. It's a long chain carbon molecules. Chemists find it very difficult to synthesize. So alternative is to get the cell biomass. So here these olinolic acid and ursolic acid, they were the isomers, and the betulinic acid. And all 3 were present in the same cell system. While compared to this, parents have only olinolic and ursolic acids. So this we have tested on the HeLa cell lines and found the anti-cancerous activity. But the cells originating from the stem had the anti-microbial activity onto it. So thank you very much. This is the IIT Guwahati campus. And
any queries? Thank you so much, ma'am. Your nice talk on plant tissue culture and its advancement is really very informative. I thank you on the behalf of whole Patanjali family. Thank you so much. जोरदार करतल ध्वनियों से आप अभिनंदन कीजिए रिस्पेक्टेड मैम का। कार्यक्रम की श्रृंखला को आगे बढ़ाते हुए हम आगे इनवाइट करते हैं डॉक्टर के राजामणि जी को। He is a professor and head of Department of Floriculture and Landscape Architecture, Tamil Nadu Agriculture University, Tamil Nadu. He has experience of 34 years in medicinal plant research and handled more than 25 externally funded projects, and in which 15 are NMPB research projects. I welcome you, sir, on the dais for the presentation.
Revered Acharya ji, all the esteemed guests, I would like to thank for this good opportunity given to me for being here, and I should thank the PRA and Dr. G.P. Rao for making this possible. So this is really a wonderful institute which I witnessed day before when I landed here. I was amazed to see this institute. I've heard about the Patanjali University. Dr. G.P. Rao used to share with me, you must see the university, so kindly make it convenient to come over for this international conference. So with that message I came, astonishing I would say, and it was just reminding me when I visited Shanghai and Beijing in 2018. So that visit for purely meant for studying the TCM, traditional Chinese medicine. So those facilities, I was just imagining when this kind of the infrastructure and the scientific laboratory will come in India. So we know how Ayush is very important to us. Last 33 years I've been working in this field. And when I entered Patanjali, really glorifying institute, I would really compliment the Swamiji and Acharya ji for building this great institute. to this level. And I would say this institute is really the propeller for the Ayush sector. So when Ayush is competing with the TCM, and this kind of institutes are to be built in India. So again, my compliments to Acharya ji. And to be brief about my own institute, so I come from 115 years old university, one of the oldest agriculture universities in India, established almost on par with Pantnagar Agriculture University. So, and continuously, TNIU has been ranking as one of the best university in India. Just last month, we got the Sardar Vallabhbhai Patel Award for the best agriculture university in India. So from that institute, I'm coming. It is one of the prestigious agriculture universities in India. And how we pursue research in many crops and pertinent to medicinal crops for that matter. My presentation will be very brief. I've got not more than 20 slides, 20-25 slides. And it will be only sharing our knowledge, how we are learning from the farmers and from the industry. So this participatory research was one of the theme which we developed, which means for a crop like medicine plant. So it cannot be an unattended area. So a farmer producing a medicinal crop, unable to sell it to the market, it becomes very difficult. And every now and then we come across many problems in marketing of the medicinal plants. So with that approach, this participatory model, in the sense, we just get attached, we means the agriculture university, we just get attached to the value chain. Say a medicinal crop which is being procured by a company, so that company forms the major stakeholder. Then the agriculture university should be one of the component. Farmer is another stakeholder. So parallel to that, we have various funding agencies, the lending agencies like banks, insurance companies. All these stakeholders put together in a value chain, we call it as the participatory research. And any research should be done in the farmer's field, in the presence of the buyer or the extracting company. So with this approach, we are moving ahead for the last 10-15 years. And I'm not just going into the details, how much we are producing. Tamil Nadu happens to be one of the major states. The rough estimate of how much we are producing from Tamil Nadu from medicinal and aromatic crops. It is around 250 crores every year. So this is a mapping which we have prepared. Around 5 or 6 major crops are cultivated in Tamil Nadu, area exceeding around 20,000 hectares. So the crop which we are seeing in the picture is Gloriosa Subaba. The Kalihari, in Hindi it's called as Kalihari. So it is one of the highly cultivated crop, which means in the last 30 years we have been cultivating this high value pharmaceutical crop. So the principal alkaloid which is being extracted from Gloriosa is colchicine. So many references were made since morning. Colchicine is being used in various phytopharmaceutical preparations. It goes mainly for the gout treatment, European or the Mediterranean fever, and for the muscular therapy. So any muscular pains, any doctor will prescribe thiocolchicoside. So it is a component based on this plant. And recently, the Canada University of Toronto has published a research paper, and their discovery is that colchicine-based drug has been developed to treat COVID. So after post-COVID, tablet in the form of tablet, it has been discovered. It's one of the major findings, and many other preparations like anti-cancerous drugs. So many valuable the phytopharmaceutical alkaloids are being generated from this crop. So every year we grow around the total area will be around 10,000 acres. In terms of hectares, we have 3,300 hectares. And nearly 10,000 farmers are there in Tamil Nadu who have been growing this high value crop for more than 25 to 30 years. So how we do research in this crop? See, we have many parameters when a new crop is being introduced. So for which there won't be any basic knowledge to the farmers available on propagation, nutrients, what is the requirement of the crop, and what is the kind of the pollination. So all those minimal requirements, we just went into the field, and as on-farm research, we proved all these technologies, and these critical techniques are being adopted by the farmers, through which they are getting higher income. Like pollination is one important parameter in Gloriosa. So if you leave it as such without pollination, only 40% of the seed yield a farmer can get. So how to pollinate, what time to pollinate, what is the stage of loss. So all these technical informations we are passing on to the farmers. Similarly, the crop nutrition, so what is the nutrient uptake pattern, and what is the nutrient scheduling. So all these informations we are giving to the farmers. Then one important breakthrough technology which we evolved for the farmers was the development of microtubers, because tubers are very, very scarce, and this plant happens to be under the RET list, which means the tubers which are normally lifted from the wild and traded to the farmers as starting material or planting material. So the resources are coming down every year because of the large-scale exploitation. To stop the wild harvest of the tubers, TNAU came out with this technology. So from the seed, we have developed the microtuber technology, through which the cost-effective planting material are generated continuously and on a sustained basis. So this is the seed, the economic part the farmer is selling to the buyers. And here again, the cumbersome method of harvesting and threshing, processing operations. We invented one small scale, a movable, portable threshing equipment. And today, as of now, nearly 1,000 units have been prototyped, and it is available to the farmers. So again, the second crop which we work, still we have the participatory research model is the Padancheru, the Coleus foscoli, tubers of which are being used for the treatment of obesity. So this again involving the main stakeholder who is buying the Coleus foscoli in Bangalore. We had an MOU with the company. They funded for the research project for 5 years, and we worked with them and developed a very unique genotype, a unique clone which has got high foscolin content. So that was released as an improved variety, and it is available in the farmers' field. So this is being marketed world over, and Japan is one of the major marketing country. So we did many analysis, genetic variability analysis, and then molecular mapping, all those parameters. This was a variety. It is a very high yielder and coupled with high alkaloid content. Similarly, the Senna, again, Tamil Nadu was the leading state to grow Senna. Now Rajasthan is also very closely following it. So in this crop also, we have more than 20 companies who are regularly buying the leaves of Senna. They buy the leaves and dry them, just pack it, and they export to the European countries. So there are many ethical issues now confronting now. Mostly the aflatoxin related problems are figuring now. So for which one National Medicine Plans Board research project, we have developed one very effective solution for the management of the aflatoxin issues. Similarly, periwinkle, Tamil Nadu happens to be the leading state in growing the Catranthus roseus. This is only alkaloid, which is very effective for the treatment of the blood cancer, leukemia. Even after 30, 40 years, there is no, there is no alternative for this. Some of the few crops which we are working very recently is one Java tea. Normally, this is one of the highly consumed tea. If you take the countries like Malaysia, we consume the black tea, they have the habit of consuming this orthosiphon stamineus. This belongs to the family Lamiaceae, the same Tulsi group. So this is the farm which is very close to Salem, and nearly 100 acres have been extended, and very high valuable cropping is being now taken up. In 1 acre, farmers are getting roughly around 1 to 2 lakhs in 1 year. This is a continuous yielding crop year after year. Every third, fourth month, they will go for the leaf harvest. So here again, we are working with the farmers, just like the Gloriosa superba. Then the next crop again, I think this is the final slide, Revolvia tetraphylla. I think you might have heard about Revolvia serpentina. Roots are being the principal source of alkaloids. But tetraphylla, there is another species in Revolvia. The leaf is being consumed. It is the raw material for extraction of many valuable alkaloids. One of the recent finding is that it yields very precious metabolites like yohimbine. So yohimbine, you might have heard, for the stamina, it is being prescribed. So this is being promoted in Coimbatore region, and in this crop also we are working with them on the standards of harvesting and how to improve the alkaloid content. So again, a few other crops like Cissus quadrangularis, and one particular unit in Tamil Nadu near Virudhunagar, single farm itself is exporting 1,500 tons of Cissus quadrangularis dry stem every year. One UK-based doctor is buying, and he converts the total raw material into drug or nutraceuticals, and it is being prescribed for the patients, especially to build the stamina, muscles, and the toning of the muscles. Like that, we have rosemary, then vetiver. With the CSIR, we also, CSIR signed an MOU with TNAU. TNAU was part of the Aroma mission, and we took part in promoting the varieties of CMAP, especially this Dharini and many other short duration varieties in Tamil Nadu. So vetiver oil, I think you should be knowing, 1 kilogram of vetiver oil, essential oil, is worth about 30,000 rupees. It's a very expensive oil, and Tamil Nadu growers are growing on a very large area. And the last slide is on Palmarosa. So we have nearly 5,000 hectares in Tamil Nadu, from which geraniol is extracted from the Palmarosa oil and is being used for many the cosmetic applications, fragrance and other applications. So I think these are the participatory research model we have, and we are very glad and to collaborate with the big institutes like Patanjali. When it comes to the raw material, we have sufficient knowledge in handling the raw materials. You name anything, any pest, any disease, and of late, many European buyers are coming, and they are approaching the universities, especially a buyer in India. He cannot export a raw material without the support of local agriculture university. In the sense, many certification issues are there, like URIGAP, the GAP, Good Agricultural Practices certifications are there, for which an agriculture university, the local university support is very much required. So I would really assure Acharya ji that TNAU will be very glad to associate with Patanjali University for any technical specifications related to medicinal aromatic plants. Thank you so much.
Any queries from the audience? So I thank you so much, sir. I appreciate your work on medicinal plants, and I think it's marketing as herbal products that you have already done is a wonderful idea. And from TNU, we have already seen a lot of research on agriculture has already been done, and surely in near future we will seek some collaboration from TNU. And on behalf of whole organizing committee from Patanjali family, I wish you a very thank you so much. इसके बाद मैं आमंत्रित करती हूँ पूज्य स्वामी परमार्थ देव जी को, जो कि पतंजलि विश्वविद्यालय के माननीय कुलानुशासक और पतंजलि के मुख्य केंद्रीय प्रभारी हैं, जिनके हृदय में पूज्य स्वामी जी व पूज्य आचार्य श्री से अनुप्राणित ऊर्जा व श्रद्धा है। मैं आपकी जोरदार करतल ध्वनि से स्वागत करती हूँ पूज्य स्वामी परमार्थ देव जी महाराज का।
वेदाहमेतं पुरुषं महान्तं आदित्यवर्णं तमसा परस्तात् तमे विदित्वाति मृत्युमेति नान्या पंथा विद्यते अनाय। ओम शांति शांति शांति। योग ऋषि परम पूज्य श्री स्वामी जी महाराज एवं हम सबके प्रेरणा स्रोत अहर्निश अखंड प्रचंड पुरुषार्थ के प्रेरक परम पूज्य आचार्य श्री जी महाराज की इस प्रेरणा को प्रणाम करते हुए, उनके चरणों में कोटिसा प्रणाम करते हुए और इस अंतर्राष्ट्रीय कार्यक्रम में जो अपने अपने क्षेत्र के विद्वान विदुषिया यहाँ पर हैं, मैं उनको भी हृदय की गहराइयों से प्रणाम करता हूँ और अपने विषय पर आता हूँ। संसार में सभी प्राणी सुख शांति से, आरोग्यता से जीवन व्यतीत करना चाहते हैं। उसके लिए परम उपदेशक, परम प्रेरक, कल्याणी वेदवाणी जो जन जन को सुख शांति और आनंद देने वाली है, उसी का जो अंतिम उपदेश है, अंतिम भाग है उपनिषद। तो उपनिषद विद्या सर्वोपरि विद्या है जो आत्म कल्याण से राष्ट्र कल्याण और विश्व कल्याण के लिए इस संसार में जानी जाती है। उपनिषदीय योग विद्या से हम शारीरिक, मानसिक, सामाजिक एवं वैश्विक समस्याओं के समाधान के लिए इसके उपदेश, आदेश, निर्देश को यदि आत्मसात करेंगे तो निसंदेह हम अपने जीवन में जो श्रेष्ठतम है, उत्तमोत्तम है, उसको हम प्राप्त कर सकते हैं। उपनिषद विद्या निसंदेह दर्शनों, स्मृतियों और जो हमारी वैदिक ऋषि परंपरा रही है, उनमें उपनिषदों का विशेष महत्व रहा है। और हमारे ऋषियों ने जो मर्यादाएं सुनिश्चित की थीं विधि और निषेध के रूप में, उस विधि और निषेध को यदि हम आत्मसात करते हैं तो आज जो वर्तमान में पूरे संसार के सामने विकट संकट मंडरा रहा है, तो शायद इससे हम अपने इन संकटों को भी दूर कर सकते हैं। अध्यात्मय जीवन, योगमय जीवन, कल्याणमय जीवन और इस जगत का जो स्पष्ट उपदेश हमारे उपनिषदों में प्राप्त है, हम यदि दृष्टिपात करते हैं आज की समस्याओं के विषय में, तो वर्तमान में विभिन्न समस्याओं के बिंदु यहाँ पर मैं आपके समक्ष प्रस्तुत कर रहा हूँ, क्योंकि समस्या सबसे पहले अपने आप में हमारे अंदर होती हैं। यदि हम व्यक्तिगत समस्याएं निपटा लें तो शायद व्यष्टिगत समस्याएं भी दूर हो सकती हैं। तो व्यक्तिगत समस्याएं अविद्या, अहंकार, प्रमाद, आहार, विचार, व्यवहार, आत्म प्रतिकूल आचरण, अब्रह्मचर्य, विषयासक्ति है। सामाजिक समस्याओं की चर्चा करें तो नैतिक मूल्यों का ह्रास, माता पिता, गुरु, आचार्य और बड़ों का अनादर। इसी प्रकार से समाज में बढ़ते हुए दुर्व्यसन, अकर्मण्यता, पुरुषार्थहीनता, ये सामाजिक समस्याएं हैं। वैश्विक समस्याओं में हिंसा, आतंकवाद, पर्यावरण प्रदूषण एवं जलवायु का असंतुलन तथा बढ़ते हुए रोगों का प्रकोप, इन समस्याओं की यदि हम समूल इनको नष्ट करना चाहते हैं तो निसंदेह इनसे पार जाने के लिए हमको ऋषियों की शरणागति प्राप्त करनी पड़ेगी। व्यक्तिगत समस्याओं का उपनिषदीय समाधान क्या हो सकता है? तो विद्या संपन्न जीवन। जिसके जीवन में विद्या है, जिसके जीवन में ज्ञान है, वही व्यक्ति सावधान है और वह इंसान नर से नारायण बन सकता है। क्योंकि ऋषियों ने कहा कि विद्या सब धनों में प्रधान है और अविद्या सभी क्लेशों का मूल है। यदि विद्या से युक्त जीवन जिएंगे तो यहाँ पर ईशोपनिषद में कहा, विद्याम च यस्तद वेदो भयम सह अविद्या मृत्युम तीर्थवा विद्यामृतम अश्नुते। यदि हम अमृतत्व की प्राप्ति करना चाहते हैं, सुख शांति आनंद के साथ जीवन को व्यतीत करना चाहते हैं, तो निसंदेह हमको विद्या से युक्त जीवन व्यतीत करना पड़ेगा। अविद्या जो अंधकार है, जो मिथ्या ज्ञान है, जो भौतिक ज्ञान है, जो केवल भौतिकता की ओर ही अपने आप को आगे बढ़ाते हैं, वे आत्म कल्याण को नहीं प्राप्त कर सकते हैं। इसलिए हम विद्या से युक्त जीवन जिए और अहंकार प्रमाद को दूर करने के लिए हमको सतत उस परम पिता परमेश्वर का चिंतन करना चाहिए जो कण कण में विद्यमान है। इसलिए सबसे पहला जो वेद मंत्र, यजुर्वेद का 40वां अध्याय, ईशोपनिषद का पहला मंत्र कहा, ईशावास्यमिदं सर्वम यत् किंच जगत्याम जगत तेन त्यक्तेन भुंजीथा मा गृधा कस्विद् धनम। इस संसार में जो कुछ भी जड़ चेतन चर अचर है वह उस परमपिता परमेश्वर के द्वारा आच्छादित है। कण कण में वही विद्यमान भगवान है। हम इस संसार में आते हैं क्योंकि जगत्याम जगत यह परिवर्तनशील है, अत्यंत तीव्रता के साथ इसमें परिवर्तन हो रहे हैं। हम अहंकार ना पालें क्योंकि अहंकार हमको गर्त की ओर ले जाएगा। हम हर क्षण इस बात का ध्यान रखें कि हमारा जो जीवन है और यह जगत है उस परमात्मा के द्वारा दिया हुआ है। अहंकार किस बात का? तेन त्यक्तेन भुंजीथा मा गृधा कस्विद् धनम। त्याग पूर्वक हम जब जीवन जीते हैं तो वह समष्टि के लिए भी कल्याणकारी होता है, उसमें व्यष्टि भी समाहित है। एक कदम जब हम पीछे से उठाते हैं तभी हम एक कदम आगे बढ़ पाते हैं। तो त्यागमय जीवन हमारी ऋषियों की परंपरा रही है। त्याग तपस्या से ही हम अपना और इस समाज का कल्याण कर सकते हैं। आहार की शुद्धि परम आवश्यक है, इसलिए उपनिषदों में कहा, आहार शुद्ध सत्व शुद्धि, सत्व शुद्ध ध्रुव स्मृति। यह संसार का नियम है और अनेक प्रकार से हम जानते हैं कि जैसा खाएंगे अन्न वैसा हो जाएगा मन। आहार की शुद्धि परम आवश्यक है। जब तक आहार शुद्ध नहीं होगा तब तक व्यक्ति का विचार शुद्ध नहीं होगा, उसका व्यवहार शुद्ध नहीं होगा। आयुर्वेद ग्रंथों में आहार के विषय में विशद से चर्चा की है और उसको हमको मानना ही पड़ेगा। आहार जो भोजन के रूप में, अन्न के रूप में लेते हैं और इन इंद्रियों से जो कुछ भी हम ग्रहण करते हैं वे भी आहार के रूप में। इसलिए हमारा सोचना, विचारना, उठना बैठना, ये सब श्रीमद् भगवद गीता में भी कहा, युक्ताहार विहारस्य युक्त चेष्टस्य कर्मसु युक्त स्वप्नावबोधस्य योगो भवति दुःखः। यदि दुखों से दूर होना चाहते हैं तो आहार विचार व्यवहार पर पूरा हमारा नियंत्रण होना चाहिए। प्रमाद ये बहुत ही बड़ा शत्रु है हमारा। आलस्यम ही मनुष्याणाम शरीरस्थो महारिपु। परम पूज्य श्री स्वामी जी महाराज भी बार बार इस बात को कहते हैं कि हम अपने प्रमाद को यदि दूर कर दे तो शायद हमको संसार में कुछ भी अप्राप्य न रहे, हम जो चाहे वो प्राप्त कर सकते हैं। इसलिए उपनिषदों में बार बार हमारे ऋषियों ने मा प्रमदः न प्रमदितव्यम इसका उपदेश किया है। तो व्यक्तिगत समस्याओं में हम प्रमादो मृत्यु कहा है, प्रमाद से हम बचें और इसके साथ आत्मानुकूल आचरण, आज विशेषकर युवा में, युवतियों में और जो भी इस समाज के घटक हैं, आत्म अनुकूल आचरण ना करने से दुख पा रहे हैं। अनेक प्रकार के अवसाद, अनेक प्रकार के रोगों से ग्रस्त हो रहे हैं और आत्महत्या करने के लिए भी विवश हो रहे हैं। इसलिए ईशोपनिषद का यह मंत्र हमको प्रेरित करता है, असुर्या नाम ते लोका अंधेन तमसावृता तास्ते प्रेत्यापि गच्छन्ति ये के चा आत्महनुजनः। हमको कभी भी आत्म प्रतिकूल आचरण नहीं करना चाहिए, वेद प्रतिकूल, वेद विरुद्ध आचरण नहीं करना चाहिए। हम आत्म अनुकूल आचरण करें। श्रूयताम धर्म सर्वस्वम श्रुत्वा च अवधारेताम आत्मनः प्रतिकूलानि परेशान न समाचरेत। यदि हम आत्म अनुकूल आचरण करेंगे तो स्वतः ही हम स्वधर्म का, स्वकर्म का पालन अच्छी प्रकार से कर पाएंगे। चौथा बिंदु है ब्रह्मचर्य से युक्त संयमित जीवन। यदि ब्रह्मचर्य से युक्त मर्यादित, संयमित, व्यवस्थित हम जीवन जीते हैं तो स्वतः हमारा जीवन प्रामाणिक हो जाता है, हम स्वतः भी निरोगी रहते हैं, हमारा घर परिवार समाज भी निरोगी रहता है। इसलिए ऋषियों ने ब्रह्मचर्य के ऊपर बहुत ही बल दिया है। वेद, उपनिषद, दर्शन और वैदिक साहित्य, लौकिक साहित्य में भी ब्रह्मचर्य के ऊपर बहुत चर्चा की गई है। इसलिए निरोगी रहने के लिए, सदा सर्वदा प्रसन्न चित्त रहने के लिए, आनंदित रहने के लिए जो मर्यादा ऋषियों ने बनाई है उसका पालन हमको अवश्य ही करना पड़ेगा। और सामाजिक जो समस्याएं हैं उन समस्याओं में नैतिक मूल्यों का ह्रास, तो नैतिक मूल्यों का हम जब पालन करेंगे, नैतिक मूल्यों को धारण करेंगे, मनसा वाचा कर्मणा, जो हमारे पूर्वजों ने जिया है, ऋषिणाम चरितम सत्यम महाजनो येन गता स पंथा, उस पथ पर अग्ने नए सुपथाराय, यदि हम सुपथ से चलेंगे, कल्याण के मार्ग से चलेंगे, तो अपने सामाजिक समस्याओं को हम स्वतः ही नष्ट कर देंगे, उन समस्याओं से हम निजात पा लेंगे। इसलिए आचार्य हमको बताते हैं, सत्यम वद धर्मम चर स्वाध्यायन मां प्रमदः आचार्य प्रियम धनम आहृत्य प्रजातंतु मां व्यच्छेत्सी, सत्या न प्रमदितव्यम, धर्मा न प्रमदितव्यम, कुशला न प्रमदितव्यम, भूत्यै न प्रमदितव्यम, स्वाध्याय प्रवचनाभ्याम न प्रमदितव्यम। ये जो उपदेश हैं ये हमारे जीवन और जगत के कल्याण के लिए अतिशय आवश्यक हैं। इसी प्रकार से माता पिता गुरु आचार्य का प्रिय आचरण भी हमको सामाजिक बुराइयों से बचाता है। यदि हम अपने माता पिता की आज्ञा का पालन करते हैं, यदि अपने गुरु की आज्ञा का पालन करते हैं, उनका प्रियाचरण करते हैं, उनके उपदेश आदेश संदेश को शिरोधार्य करते हैं, तो उससे स्वयं भी और इस समाज का भी हम कल्याण कर पाते हैं। इसलिए कहा मातृदेवो भव, पितृदेवो भव, आचार्य देवो भव, अतिथि देवो भव। ये जो हमारे संसार के कल्याण के लिए ऋषियों ने बताए मार्ग पर हमको चल करके निसंदेह सामाजिक और व्यक्तिगत समस्याओं से हम निजात पा सकते हैं। बहुत बड़ी समस्या व्यसनों की है। आज संसार में कहीं भी हम जाएं, अलग अलग प्रकार के व्यसनों से युक्त व्यक्ति और समाज को हम देखते हैं, जिससे घर परिवार टूटते हैं, जिससे दिलों में दरारें पड़ती हैं, दीवारें खड़ी हो जाती हैं और घर बर्बाद हो जाते हैं। अलग अलग प्रकार के रोग दोष से व्यक्ति युक्त हो जाता है। इसलिए हम यदि उपनिषदों का उपदेश सुनते हैं, पढ़ते हैं, तो उनका उद्घोष हमको स्मरण रखना चाहिए। जहां पर उद्घोष एक राजा करता है कि मेरी प्रजा कैसी है, नामिस्तेनो जनपदे न कदर्यो न मद्यपो नाना हिताग्नि अविद्वान स्वैरी स्वैरिणी कुतः। ये संसार का नियम है कि भारत वर्ष का यदि इस ऋषि परंपरा का यदि किसी ने आचरण किया है तो उसका स्वयं का जीवन, उसका व्यक्तिगत, सामाजिक और वैश्विक उन्नति सदा सर्वदा होती रही है। वेदोक्त कर्म, यदि हम वेदोक्त कर्म करते हैं, अनासक्त भाव से कर्म करते हैं, निर्लेप भाव से कर्म करते हैं, तो सामाजिक समस्याओं से हम निसंदेह मुक्त हो सकते हैं। इसलिए कहा कुर्वन्निवेह कर्माणि जिजी विशेषतम समाह एवं तो नान्यथेतोस्ति ना कर्म लिप्यते नरे। जब हम वेदोक्त कर्म करेंगे तो निसंदेह इन बीमारियों से, इन रोगों से, दोषों से बच पाएंगे, अहंकार से बच पाएंगे। विश्व की अब समस्याओं पर चर्चा करें तो हिंसा, आतंकवाद की समस्या, पर्यावरण प्रदूषण की जो समस्याएं हैं उसका असंतुलन है और बढ़ती बीमारियों का जो प्रकोप है, इसमें भी ऋषियों का उपदेश सिरोधार्य है। वेदोक्त अहिंसा का पालन, सभी प्राणियों के प्रति भागवत प्रेम, आत्मवत व्यवहार। यदि हम आत्मवत व्यवहार करते हैं, यदि हम किसी से घृणा नहीं करते हैं, सब में उस परमपिता परमेश्वर का स्वरूप देखते हैं, तो यस्तु सर्वाणि भूतानि आत्मन्यवानुपश्यति सर्वभूतेषु चात्मानं ततो न विजुगुप्सते। यस्मिन् सर्वाणि भूतानि आत्मैव भूत विजानतः तत्र को मोहा कः शोका एकत्वम अनुपश्यता। यत्र विश्वम भवत्येकनीडम, जो उद्घोष वेदों का है, जो ऋषियों का है, इससे हम आज के जो विकट संकट में, जहां पर मजहबवाद है, जहां पर प्रांतवाद है, जहां भाषावाद है, जहां पर एक जो पिपासा है, जो भौतिकता है, जो अहंकार की पुष्टि के लिए लोग एक दूसरे पर आक्रमण कर रहे हैं, एक दूसरे का हक छीन रहे हैं, एक दूसरे का गला रेत रहे हैं, तो वहां पर यदि हम इन उपदेशों को ग्रहण करके आत्मत व्यवहार करते हैं तो निसंदेह वैदिक परंपरा से हम इस विश्व को बहुत सुंदर बना सकते हैं। यज्ञमय जीवन, क्योंकि यज्ञो वै श्रेष्ठतमम कर्म, यज्ञ जो बहुत ही वैज्ञानिक है, जहां पर पर्यावरण प्रदूषण अधिक है, रोगों का प्रकोप अधिक है, वहां पर यज्ञ करके बहुत रिसर्च इस पर हो चुकी है, निरंतर हो रही है। और पतंजलि योग पीठ के माध्यम से भी परम पूज्य आचार्य श्री जी महाराज, श्री स्वामी जी महाराज के पावन निर्देशन में रिसर्च का कार्य निरंतर चलता रहता है। तो हमारे ऋषियों ने, ऋषि रिसर्चर भवति, ऋषियों ने रिसर्च किया और पाया कि यज्ञ यदि हम करते हैं तो उससे आध्यात्मिक, भौतिक और सब प्रकार के सुखों का, आनंद का हमारे जीवन में आधान होता है। योगमय जीवन, आज अलग अलग प्रकार के रोगों का आक्रमण हो रहा है, छोटे बच्चे से लेकर के वृद्ध तक नाना प्रकार के रोगों से निरंतर लड़ते रहते हैं। वहां पर योग, आयुर्वेद, तो योग के विषय में श्री स्वामी जी महाराज ने अपने अखंड प्रचंड पुरुषार्थ से, अपने तप से आज दुनिया के कोने कोने में ऋषि परंपरा का बहुत बड़ा कीर्तिमान स्थापित किया है। उन्होंने घर घर योग पहुंचाया है और ऋषियों का यही कथन है, उनका उपदेश है, प्राणस्थिदम वसे सर्वम त्रिदिवीयत प्रतिष्ठितम मातेव पुत्रान रक्षस्व श्रेष्ठ प्रजाम च विदेहि नैति। और जो वास्तव में इस प्रकार से योगमय जीवन जीता है वही तन, मन और वतन का आरोग्य स्थापित करता है। न तस्य रोगो न जरा न मृत्यु प्राप्तस्य योगाग्निमयम शरीरम की यह भावना अत्यंत उदात्त है और इस परंपरा का हम सब निर्वहन कर रहे हैं। पतंजलि योग पीठ में परम पूज्य आचार्य जी और महाराज श्री का जो संदेश हम सब आत्मसात करके इस उपनिषदीय विद्या का, योग विद्या का, आत्म कल्याण से विश्व कल्याण के मार्ग में हम सदा इसको धारण करें और इससे हमारी संस्कृति का गौरव बढ़ेगा, इसमें कोई संदेह नहीं है। पुनश्च मैं हृदय की गहराइयों से परम पूज्य आचार्य श्री जी महाराज के चरणों में, परम पूज्य महाराज श्री के चरणों में, आप सभी विद्वानों के चरणों में नमन करता हूँ, प्रणाम करता हूँ। ओम इति शम।
बहुत बहुत धन्यवाद पूज्य स्वामी जी, आपका मार्गदर्शन पाकर हम लोग धन्य हैं। मैं हृदय की गहराई से नतमस्तक होते हुए आपका हार्दिक वंदन और अभिनंदन करती हूँ। ये एक छोटा सा प्रसंग आप कह सकते हैं कि योग का, क्योंकि विश्वविद्यालय का कार्यक्रम है, विश्वविद्यालय के कार्यक्रम के तहत ही पूज्य स्वामी जी का कुछ हमें इसके प्रति मार्गदर्शन प्राप्त हुआ। अगला कार्यक्रम मैं आमंत्रित करना चाहूँगी डॉक्टर टी शेखर जी को। He is the professor in Department of EVS, Igantu, Amarkantak, Madhya Pradesh. He is having 27 years of teaching experience. His area of specialization is plant anatomy, histochemistry, biodiversity, and in vitro conservation of medicinal plants. I welcome you on the dais, sir, for the presentation. Thank you.
Namaskar. At the outset, I thank the organizers, Swami Ramdev ji, Acharya Balkrishna ji, and Professor Bhatnagar, Professor G.P. Rao, and team, for having given me the opportunity to present before you some of my findings on the conservation side of aspects of the medicinal plants for the past 12 years, which some of my students have worked on that, and with the support of National Medicinal Plan Board for the past 1 decade, we have conserved more number of RET species because conservation, since last 2 days, many speakers spoke about the need for medicinal plants, its utilization, its validation, its phytochemicals, and its applications for various human diseases, and so on. So for which, so where is the raw material? So where is the raw drugs? Where do you get from that? Some of them are wild, some of them are cultivated, and how to protect that from the natural forest. So we focused only on the conservation of RET species in our laboratory, and we worked many species which is prevailed on the Western Ghats, especially from Tamil Nadu to Karnataka, Kerala, the 3 states, that belt we covered. And my previous speaker, madam, spoke about the plant tissue culture and its applications. 3 types of applications: qualitative, quantitative, and secondary metabolites. And we have chosen that qualitative first type, how to conserve it in in situ conservation of the RET species. And it is being much exploited by the traders, much exploited by illegal means. And most of the population is at the verge of extinct, already endangered, that is already RET, and some of them are endemic as well. So the natural resources should be protected. And who is to conserve? And who is responsible? We are all responsible, especially for young students, researchers, public, government side. We not depend for all this purpose to the government side to conserve or protect the genotypes. And we all should take responsibility. Like we plant trees every day. Plantation of trees in all functions, all occasions, all VIPs they plant, massive plantations are there to make the environment more green. So likewise in the medicine plant sector, if you want this field to sustain, if you want a raw material, for which you should have a quality planting material. So where do you get the quality planting material? Unless it is being protected from the natural resource, its own habitation in the forest, as well as the great institution like Agriculture Research Institutes, and as well the Patanjali and leading universities, they should take up the issue for the conservation aspect. And Swamiji was telling here, the herbal garden is there. See, through the herbal garden, we have a sampling plants to create awareness for the common people and students, and for the raw material for research R&D purpose only. When you go for large scale cultivation, we need a lot of raw drugs. For the phytopharmaceutical industry, now it is being focused. The recent science, Ayurveda and Siddha are focusing. The government of India is pumping a lot of funds on this area to popularize the indigenous medicine to cure a lot of ailments to the humankind in the great motherland. So in a way that, as I told you, the conservation aspect, and we have taken, small introduction, all you know about it because of time constraint. I need not read all this. And for the medicinal plant species recorded from different countries and regions as well, China stood first, India followed by it, total number of plant species, number of medicinal plants, and 42% of Indian country's contribution. Ethnobotany and ethnomedicine or some basis, you need not, and some of the botanical drug discovered based on the traditional medicine, or the common, very important medicinal plants and common diseases and the active compounds which are present. And India is one of the 12 mega biodiversity centers, or 18,000 flowering plants, more than 6,000 medicinal plants are present, rich for traditional knowledge, rich heritage and herbal medicine, large biodiversity, which lead world market due to export of crude drugs extracts. Here various types of traditional and modern systems of medicine in India, with the tribal 7,000, Siddha 700, and modern medicine, Unani, Tibetan and Ayurveda 2,200. And major Indian medicinal plants used in Ayurveda, Siddha, Unani systems of medicine, few of them I listed, which from the literature, most commonly used medicine which is being exploited in the three system of medicines in our country. Major medicinal plants exported from India, some of the list, and its parts of the plants, the root, rhizome, etc. And plant diversity in Tamil Nadu. Tamil Nadu is southern tip of rich in flora and fauna, Western Ghats, Eastern coastal plains where a lot of plants diversity is rich, high endemism is there. And Tamil Nadu harbors more than 5,200 species of plants, more than thousands of medicinal plants. Again, the conservation of medicinal plants. There is a threat to medicinal plants by means of increasing usage level, including industrial use and collection. Villages use them for the home remedies, collection by traditional practitioners, practice of rituals and collecting requirements, and commercial collectors, increasing exports, destructive harvesting techniques, deforestation, and habitat destruction are some of the means we need to. There is a threat for the medicinal plant survival, for which we have to go a long way, and we took much care to the conservation of the medicinal plants. Who to conserve and what to conserve? The endemic species are to be conserved. Species for which therapeutic efficacy it proves should be conserved. Species of ethnomedicinal importance should be conserved. Species which are most important are third to conserve. And conservation of medicinal plants, conservation of in natural environment, and conservation in aligned to the environment, ex situ conservation and in situ conservation, and biotechnology in conservation, which is used as a tool to conserve the plants by means of cryobanks, DNA banks, and in vitro multiplication, so on. Objective of department, as I told you, we have concentrated on the conservation of the RET plants. And these are some of the list of plants in the laboratory. We have conserved through the plant tissue culture technology, the in vitro conservation, lab to land technology, and we have done more than 24 species, some of them which I am going to highlight here. This is the micropropagation, what we developed a protocol from the leaf-wax plants through calendinogenesis for the Artemisia annua, and the ardent plants will be transferred to the field and supplied to the farmers for the cultivation. And next one is Decalipus hamiltoni, is very important medicinal plants. Here nodal explants were used for the direct development of plantlets without calendinogenesis. In this protocol also we have developed and supplied the plants to the local farmers to cultivate. And in vitro shoot generation of Hexacum and Justicia genderosa, Nothopoditis pneumoniana or Fotida. Here this is very important anti-cancerous plant which is prevalent in the Nilgiris. We have developed through the leaf and Plumbago rosea, Ruta graveolens, Stevia rhodiana, Tylophora indica, Vanilla planifolia, and so on. So all this, some of our contribution for the conservation inside the in situ conservation protocol we have developed, and we have generated a lot of seedlings. To conserve the commercially important and threatened medicinal plants, the in vitro propagation is important tool for in situ conservation. So there is urgent need to develop standard tissue culture protocols for the conservation of such valuable medicinal plants. So this is about the conservation aspect. Next, our own personal experience in my native village. As Professor Rajamani was telling, the participative program, a lot of medicinal plants the TNAU has been advised the farmers to cultivate for their livelihood, in which we belong to Salem district of Tamil Nadu, where we have taken up some of the plants for our the cultivation for the livelihood of the local farmers, and this is about some of the details about the herbal medicine plants and their therapeutic uses, and cultivation of Coleus forscoli. We have taken the field of our own farm in the Salem district, and this is a field which is leveled, and we have prepared. The Coleus is being cultivated, and it is well grown plant in our field. See the root biomass which is being harvested, dried in the yard, and is being marketed to the Sami laboratory which is near to Tamil Nadu, Bangalore, and we have that person have a tie-up with the farmers, and he is helping the farmers for the trading purpose, and so this kind of the cultivation of the medicinal plants will support the livelihood of the rural farmers to enhance their financial status to survive in the day-to-day life. So with this I conclude my talk. Thank you so much for the opportunity.
Any queries? Thank you so much, sir. We are indebted for your precious time and knowledge sharing ideas. After this conversation, I invite on the stage Professor Sangeeta Saxena ji. She is the professor and head in Department of Biotech, Baba Saheb Bhim Rao Ambedkar University, Lucknow. Her specialization is in plant molecular biology, molecular virology, RNA interference, molecular markers, and cross-kingdom regulation of plant miRNA. I welcome you on the dais, ma'am. I hope your illuminating talk will enlighten the ideas of our budding scientists. Thank you.
Thank you very much, Priya ma'am. Pujaniya Swami ji, Acharya ji, Professor Bhatnagar, Professor Rao, Professor R.P. Singh, eminent scientist, faculty, research scholar, and dear students. So first of all, I would like to say thanks to the organizers for giving me this platform to present my views in this very significant conference, international conference on medicinal plants. सबसे पहले मैं PRI के युवा वैज्ञानिकों, पतंजलि यूनिवर्सिटी की फैकल्टी और छात्र छात्राओं से कहना चाहती हूँ कि आप लोग बहुत किस्मत वाले हैं। आपका प्रांगण बहुत पवित्र है, बहुत सुंदर है, आपकी आप भगत बहुत निराली है, और आज सुबह स्वामी जी आचार्य जी के सानिध्य में जो आशीर्वाद हम लोगों को प्राप्त हुआ, वो बेहद सुखद अनुभव है हम सब के लिए। इस तरह की कॉन्फ्रेंस तो हम लोग अक्सर अटेंड करते हैं, पर इस किस्म का अनुभव हमें पहली बार हो रहा है और बहुत अच्छा लग रहा है। हम लोग सच में बहुत प्रसन्न हैं। तो धन्यवाद आचार्य जी, स्वामी जी। So coming to my topic, I am going to speak today on bioactivity of Carica papaya leaves extract for increased life expectancy and delayed aging. So I would like to give you some background that we have been working in the area of plant viruses for very long. So we were the first one to identify papaya leaf curl virus to be a Gemini virus, and we completely sequenced its genome. And then nowadays we are doing management of this disease by technology which is called as siRNA or gene silencing using small interfering RNA. जब हम लोग ये काम कर रहे थे, तब हम लोगों ने सोचा कि क्या हम बायो एक्टिविटी कैरिका पपाया लीव्स की देख सकते हैं हेल्दी एज कम्पेयर टू वायरस इन्फेक्टेड। सो आप में से बहुत सारे लोग कहेंगे कि वायरस इन्फेक्टेड प्लांट्स तो हम रोज़ खाते हैं। एक्चुअली हम जो भी खाते हैं उसमें ज़्यादातर प्लांट्स में वायरस होता है, चाहे मूली खाएं, गाजर खाएं, टमाटर खाएं, दे ऑल आर इन्फेक्टेड विद plant viruses, but they do not harm us. हमें डरने की ज़रूरत नहीं है. Plant viruses हमको कोई नुकसान नहीं पहुंचाते क्योंकि हमारे अंदर वो रिसेप्टर्स ही नहीं होते जिन पे प्लांट वायरस बाइंड करता है. जैसे कोरोना वायरस आया था तो आपके पास ACE2 रिसेप्टर था. अगर वो रिसेप्टर ही ना होता तो शायद कोरोना हमारा कुछ बिगाड़ ना पाता. तो हमने सोचा कि हम देखें कि पपाया लीफ की हम बहुत चीज़ों में इस्तेमाल करते हैं. डेंगी में आप ही पतंजलि ने ही ड्रग बनाई, डेंगीनिल, और कैरिपेल आती है. पपाया का ऑलमोस्ट हर पार्ट खाया जाता है और किसी न किसी औषधि के लिए उसका इस्तेमाल होता है. तो हमें लगा हमें डरने की ज़रूरत तो नहीं है कि हम इन्फेक्टेड लीफ खा रहे हैं, लेकिन हमें ये देखने का में थोड़ा अच्छा लगा कि क्या इसकी औषधीय गुणों पे कोई फ़र्क पड़ता है क्या. तो ख़ास तौर पे जो जनमानस प्लेटलेट्स की कमी के लिए कच्चा पपीता खाते हैं या पत्ती का एक्सट्रैक्ट लेते हैं, क्या हम उनको अवेयर कर सकते हैं वायरस इन्फेक्टेड और हेल्दी लीफ के लक्षणों के लिए. जैसे आज सुबह आचार्य जी ने भी कहा कि हम सारी क्लासिफिकेशन, सारी टेक्सोनॉमी रट के पास नहीं कर सकते. अगर वो लक्षणों के ऊपर होंगे, उनके नाम तो शायद हमें ज़्यादा आसानी से याद होंगे. ऐसे ही अगर हम जनमानस तक पहुंचाने के लिए वायरसेस के सिम्टम्स के बारे में कुछ बता सकें तो शायद वो बहुत अच्छा होगा. लेकिन उसके संग संग जो कमर्शियली प्रोपेगेट कर रहे हैं या मेडिसिनल प्लांट्स की खेती कर रहे हैं, उनके लिए एक अच्छा आइडिया है कि वो मॉलिक्यूलर टेक्निक्स को यूज़ करके वायरस को आइडेंटिफाई करें ताकि औषधीय गुणों में कोई फ़र्क ना पड़े. वैसे हमें प्लांट वायरसेस से डरने की कोई भी ज़रूरत नहीं है. तो हमने उसको सी एलिगेंस को ही मॉडल ऑर्गेनिज्म की तरह यूज़ करा. और डॉक्टर पांडे बैठे हैं हमारे बीच में. डॉक्टर पांडे के ही एक स्टूडेंट हमारे यहाँ पोस्ट डॉक बन के आए थे और 1 साल पहले ही हमने सी एलिगेंस की लैब अपने यहाँ इस्टेब्लिश करी है. तो मुझे प्लांट वायरसेस पे काम करते करते ये आइडिया बहुत अच्छा लगा कि मैं बायो एक्टिविटी चेक करूं हेल्दी और इन्फेक्टेड पपाया के लीव्स की. तो सी एलिगेंस के बारे में डॉक्टर पांडे ने ऑलरेडी बहुत कुछ बता दिया है. ये मॉडल ऑर्गेनिज्म है, लेकिन मैं एक बात आपको बताना चाहती हूँ. इसके पास 60 से 80% होमोलॉजी है ह्यूमन जीन से. ना सिर्फ ये, हम बहुत सारी म्यूटेंट सी एलिगेंस की लाइंस अवेलेबल भी हैं और जनरेट भी कर सकते हैं मॉलिक्यूलर टेक्निक से, म्यूटेंट बनाकर और क्रिस्पर टेक्नोलॉजी से, ताकि हम पिन पॉइंटेडली उसी जीन को अटैक करें जिसका हम रिवर्सल फंक्शन अपनी ड्रग या अपनी फार्मास्यूटिकल या न्यूट्रास्यूटिकल प्रॉपर्टी में देखना चाहते हैं. बाकी आपने सुबह सी एलिगेंस के बारे में डिटेल पढ़ ही लिया है. सी एलिगेंस की लाइफ साइकिल है जो आप लोग देख रहे हैं सामने, और हम इसी पे काम करते हैं. और ये क्योंकि बहुत फास्ट ग्रोइंग ऑर्गेनिज्म है और बहुत जल्दी इसके रिजल्ट्स मिलते हैं, तो हमको भी ये मॉडल ऑर्गेनिज्म अपनी लैब में रखना बहुत अच्छा लगा, क्योंकि हमारी लैब में बहुत सारा काम मेडिसिनल प्लांट्स पे करना हमने शुरू करा है और हम सी एलिगेंस को मॉडल ऑर्गेनिज्म की तरह यूज़ कर रहे हैं. सो मेनी मेडिसिनल प्लांट्स आर टेस्टेड फॉर देयर थेराप्यूटिक पोटेंशियल ऑन सी एलिगेंस. हल्दी है, बकोपा है, पपाया करते ही हैं हम लोग, और बहुत सारे हैं जो ओबिसिटी के लिए, कैंसर के लिए, एंटी हेलमेंथिक के लिए. कैरिका पपाया एक्चुअली कीड़ों के लिए बहुत अच्छा इस्तेमाल होता है, एजिंग के लिए. और बहुत सारा लिटरेचर हमें ऐसा मिला जिसमें पपाया का रोल एंटी एजिंग या डिलेड एजिंग के लिए दिखाया गया था. पपाया एक बहुत इम्पोर्टेंट फ्रूट है, बहुत इम्पोर्टेंट प्लांट है. हर पार्ट उसका यूज़ होता है, थेराप्यूटिक्स में, बेबी फूड में, कॉस्मेटिक्स में, फार्मास्यूटिकल में. बट इसका एक मेजर ड्रॉबैक ये है कि इसके ऊपर बहुत सारे प्लांट वायरसेस का प्रकोप रहता है. आई एम श्योर आपने अपने किचन गार्डन में जब कभी भी पपाया उगाया होगा, दैट पपाया मस्ट बी इन्फेक्टेड विद पपाया लीफ कर्ल वायरस और पपाया रिंग स्पॉट वायरस. So this papaya plantation is almost wiped out in whole of Uttar Pradesh because of the susceptibility to this particular virus. ये जो virus है, it's a DNA virus. Another virus, papaya ring spot virus, is an RNA virus, and we work a lot on these viruses. इनके vector होते हैं, ये छोटी सी सफ़ेद रंग की एक मक्खी होती है. अगर आप बहुत धीमे से पपीते की पत्ती को पलटें या बैगन या टमाटर या चिली को पलटें तो आपको ये वेक्टर दिखाई देगा. तो इसको मैनेज करने के लिए आप वेक्टर को भी मैनेज कर सकते हैं ताकि ये डिजीज़ आप मैनेज कर पाएँ. और बाकी मैनेजमेंट प्रैक्टिसेस तो चली रही हैं, बट आपको केमिकल छिड़कने की कतई ज़रूरत नहीं है, क्योंकि इसका उपचार यही है कि आप अर्ली डिटेक्ट करें, अपरूट करें और जला दें, ताकि इनिशियल इनोकुलम आपकी फील्ड में से कम हो जाए. अगर ये प्रैक्टिसेस मेडिसिनल प्लांट्स ग्रो करने वाले लोग करने लगें तो मुझे लगता है ये बहुत एक फ़ायदे का सौदा होगा. ये पपाया रिंग स्पॉट वायरस है और आप जितने भी पपीते बाजार से खरीदते हैं उन सब में आपने ये गोल गोल रिंग्स देखी होंगी जो रिंग स्पॉट वायरस का एक प्रतीक है, और हम इसको खाते हैं और हमें कोई दिक्कत नहीं है उसको खाने से। मैं फिर से एम्फेसाइज करना चाहती हूँ, प्लांट वायरसेस आर एब्सोल्युटली सेफ फॉर अस टू ईट, बट वी वुड लाइक टू सी द न्यूट्रास्यूटिकल और imbalance in the therapeutic potential of the infected as well as healthy plant. So we can easily identify the infected plant as compared to the healthy plant. As you can see it, this is a very I will say clear-cut example of leaf curl virus, but there are many symptoms which a common person cannot actually identify. But nonetheless, you people should be aware that these kind of symptoms are there. तो कभी आपको डेंगी में या प्लेटलेट्स कम काउंट होने पे पत्ती तोड़नी पड़े तो कोशिश करिए कि आप उस पे देखिए कि कोई सिम्टम ना हो और तब आप उसकी पत्ती लेकर उसका एक्सट्रैक्ट निकाल के पीजिए। हमने सेम किया, स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोपी करी और हमने देखा बहुत सारे डिस्टॉर्शंस हमको स्टोमेटा और लीफ की एनाटमी में मिले। हमें बहुत सारा एलिमेंटल एनालिसिस में भी बहुत सारे फ़र्क मिले इन्फेक्टेड लीफ में एज़ कम्पेयर टू हेल्दी, और हमने मॉलिकुलर प्राइमर्स बनाए और हमारी लैब में डायग्नोस्टिक किट्स दोनों पपाया लीफ कल वायरस और पपाया रिंग स्पॉट के लिए अवेलेबल है, जो मैं चाहती हूँ कि आम आदमी तो बिल्कुल नहीं लेकिन कमर्शियली जो ग्रो कर रहे हैं वो लोग यूज़ करें, सो दैट they can identify at the very early stage when symptoms are not so obvious and they can do the needful in the papaya fields. So we can identify the virus by various ways, but we prefer PCR based molecular detection of this virus. Either it's an RNA virus or DNA virus, both ways we can do it. तो हमारे पास बहुत सारे degenerate primers group specific और geography के हिसाब से isolate specific हैं, जिससे हम आपको टेस्ट करके एक बार में 100 रैंडम सैम्पलिंग आराम से टेस्ट हो सकती हैं और आप इनिशियल इनोकुलम कम करके हमेशा के लिए इसी का नहीं बल्कि किसी भी मेडिसिनल प्लांट पे, मैं अभी लिटरेचर सर्वे कर रही थी और हल्दी पे CMV बहुत आता है और पपाया पे तो खैर वायरसेस है ही हैं और विदानिया पे भी प्लांट वायरस की प्रॉब्लम है। तो आई थिंक वी शुड, प्रोफेसर राव इज सिटिंग हेयर, so I think we should do something, some database or data bank with respect to plant viruses infecting medicinal plants. That can be a very good added information or top-on information in the existing knowledge of medicinal plants. So we tested papaya leaf extract in a very simple manner, as Dr. Pandey already told. So we just crushed it and we centrifuged it and then we treated the C. elegans and we simply identified, or you can say we tried to evaluate the lifespan and lethal concentration of papaya. And interestingly what we found was that healthy papaya leaves were less lethal in C. elegans as compared to the virus infected leaves. So our LC value was much more higher in case of healthy plants. Similarly, healthy papaya leaves increased the life expectancy and delayed aging in C. elegans as expected, but the same results were not found in case of virus infected leaves, and more detrimental results were found in case of papaya plants which were infected with papaya ring spot virus. So you can see we have a clue here that the nutraceutical or therapeutic potential can be hampered if the plant, the medicinal plant which we are using for therapeutic purpose is infected with viruses. Virus is the only pathogen which multiplies inside the cell, else all the pathogens are outside. So you see, we have to be a little bit more particular when we are using or when we are talking about the viral diseases in plants or animals for that matter. So we recently published a paper, I think 2 months back, in plants with impact factor 5, where we have done all these analysis of papaya leaf curl virus on papaya, and the biological activity has been given there. So we conclude that papaya ring spot and papaya leaf curl virus may affect the nutraceutical or therapeutic potential of Carica papaya, number 1. Number 2, we are also developing an Android app and web app to help common man to identify the virus infecting papaya. So we are generating more and more pictures from different geographical locations with different symptoms from different angles to increase the confidence level of this software, and once we are done with it, probably a common man just with the click of this app on the mobile can identify that whether this papaya plant is infected with a virus or not. We may do the same thing with medicinal plants also. And I also believe that common man should be sensitized to identify it, or one should be aware of compromised nutraceutical or therapeutic property of papaya leaf extract if it is infected with papaya leaf curl virus. So this was a brief presentation, and happy birthday Acharya Balkrishna ji. Thank you very much. So
Thank you so much, ma'am.
Yeah, any queries?
Yeah, any queries? Thank you so much, ma'am. Your work on papaya is much appreciable, and we are also doing extensive research in our Drug Discovery and Development Division on C. elegans, and I again thank you so much. In continuation, I invite on the stage Dr. H.K. Pandey ji. He is the scientist in DRDO, Pithoragarh, Uttarakhand, and he has been awarded as a DRDO's Scientist of the Year award for his contribution in developing several herbal medicines, including the popular drug leucoskin. He has more than 28 years of research experience on Himalayan medicinal plants and development of herbal products. I welcome you on the dais, sir.
Honorable Acharya, dignitaries on the dais, young researcher of Patanjali, and my dear students. The Phytomedicine Division of Defense Institute of Bioenergy Research, DRDO, is doing R&D work on Himalayan medicinal plants, particularly on their conservation and value addition. Under value addition, we have developed 7 herbal products and filed 8 patents also. We have developed herbal product for leukoderma, eczema, anti-UV radiation cream, herbal health supplement, antioxidant and immunostimulant herbal food supplement, anti-toothache herbal formulation. And recently we have developed anti-leukoderma herbal product MAR2, advanced version of leucoskin. Leukoderma, as you know, it is skin depigmentation disorder. The skin becomes white due to lack of melanin. Melanin is a protein responsible for the coloration of the skin. And there is no satisfactory cure of this disease. It is almost incurable. In modern medicine or in allopathy, it is almost incurable. Doctor says you have to live up with this disorder. Some treatments are available in Ayurveda, but efficacy is very less, 10 to 15%. And it is a big social stigma in our country. The affected individual always remains in severe mental tension, being socially outcast. Keeping all these aspects, we have developed a herbal formulation for this, because etiology of the disease is very diverse. Almost 12 to 13 causes are responsible for leukoderma. We have formulated a product. It is a polycomponent herbal product containing 2 components, ointment for topical application and oral syrup. And after completing all the desired studies, we have transferred technology of this product to Emil Pharmaceutical. Emil has launched this product by the trade name of leucoskin. This product is in the market and being marketed by Amazon, Flipkart, and clinical efficacy of this product is 60 to 65%, and recovery time is 120 days to 600 days. The main biomolecules are ferronicoumarins. Product is toxicologically safe and free from heavy metals. LD50 is more than 2 grams per kg body weight, and lethal surface 50 is 1 gram per kg body weight. In last 4 years, the sale of this product has earned 2 crores 50 lakh royalty to DRDO. And still there was scope to enhance the efficacy and to reduce the time of recovery. So we have developed advanced version of leucoskin. I am going to present that. And this is the launching photograph of the product. If you look at the definition, it is the disorder of skin in which melanin biosynthesis is disturbed, either by destruction of melanocyte cells or tyrosinase enzyme inactivity, resulting white lesions on the skin. Incidence of leukoderma is 1 to 2% worldwide. In India, its incidence is 3 to 4%. But in some parts of Gujarat and Rajasthan, its incidence is almost epidemic level, 7 to 8%. Vitiligo it is called vitiligo or Svetkusta or Fulleri in Hindi. It is of 3 types: vitiligo localista. Vitiligo localized lesions are present on the skin. It may be unilateral or bilateral. With generalista, almost 40% of cutaneous body is white, and generalista almost 70 to 80% body is white. Etiology, causes of the disease, more common among darker races, particularly in the age of 10 to 30 years. 30% cases, it runs in the family, heredity also. If a person is suffering from autoimmune disorder like hyperthyroidism, adenocortical disorder, pernicious anemia, diabetes mellitus, alopecia, and psoriasis, he is prone for leukoderma. If a person is in regular contact with the chemicals like monobenzyl ether of phytoquinones, phenols, chances of leukoderma are there. Deficiency of copper, vitamin B12, and aromatic amino acids. Phenylalanine, tyrosine are also responsible. Theory of origin, chronic hepatic disorder. If some stomach regular or chronic stomach disorder also leads to leukoderma. If a person is in some industry or in regular contact with chemicals, chances of leukoderma are there. Intestinal parasitism is also responsible. Hormonal theory, lack of MSH, melanocyte stimulating hormone, because in the posterior lobe of pituitary melanocyte hormone is secreted by some shock or some mental disorder, that lobes gets affected, then it affects the secretion of melanocyte stimulating hormone, ultimately leads to leukoderma. Neurogenic theory, neurotoxins like phenol, quinones, hydroxyindols, and cuprous melatonin are deposited on the nerve endings. You might have seen leukoderma on the fingertips, nose, ear. It is due to the neurogenic theory. Autoimmune response, it is autoimmune disorder also. Body recognizes certain melanocyte cells as foreign cells and start destroying them. Infective theory, viral and mitotic infection are also responsible for leukoderma. Existing modalities, topical steroidal therapy, ointment of dexamethasone, hydrocortisone are 1 or 2% composition available in the market. Topical psoralen UVA therapy, psoralen ointment applied on the skin topically in the form of ointment, and oral psoralen UVA therapy, and followed by the exposure of UVA radiation. Surgical therapies are also there. Skin graft, skin of the normal side of the patient is taken out and grafted on the white patch site. Blister grafting, blisters are created on the normal skin as well as on the white patch and transplanted. Normal skin is transplanted on the grafted skin. Micropigment tattooing is again one of the cause. If a small patch is there, it can be done. It can be chemophilus through tattooing. Autologous melanocyte transplant. Melanocytes are cultured in the lab and transplanted on the depigmented area. Sunscreen and cosmetics. Cosmetics are also Lydia and Dermaband skin colors are available in the market, available with internet or Amazon, and just that it can hide, these pigment can hide the white patch. These are the drawbacks. The topical steroidal therapy is restricted on the localized lesions, mostly on the eyes, face, but it causes shrinking of the skin, stain on the skin. Topical UVA therapy, it causes Stevens-1 blister formation. Oral UV therapy, again, severe sunburn, nausea, vomiting. Surgical therapy, all these surgical therapies are experimental and restricted to the localized lesions only. No answer when the disease is progressing. Overall, these therapies are, efficacy of these therapies are very less, 10 to 15%. This is the biosynthetic pathway of melanine. It starts from the aromatic amino acid phenylalanine, and after 8 steps, it becomes indole 5,6 quinone, which polymerizes into the melanine. Because the main enzyme is tyrosinase, and copper is the prosthetic group of that enzyme. In what we did in Mark II, we have made changes in the ointment as well as oral dose to enhance the efficacy and to reduce the time of recovery. 7 ingredients of 7 plants are there in the ointment, and ingredients of 6 plants are there in the oral dose. The main biomolecule in anti-leukoderma herbal product Mark II are furanocoumarins. 4 types of furanocoumarins are there in our product: sorolin, isosorolin or angelicin, 5-methoxysorolin, and 8-methoxysorolin. In ointment as well as oral dose, 8-methoxysorolin was found in maximum quantity, followed by isosorolin. This is the HPLC chromatogram of ointment as well as oral dose. This is the HPTLC fingerprint of the product, ointment and oral dose. Micro elements play a major role in the pigment formation, particularly copper, because copper is the prosthetic group of enzyme tyrosinase, and the main enzyme responsible for the coloration of the skin or melanogenesis. And in our product, availability of ointment as well as oral dose, availability of copper, zinc, and iron was in ample quantity. These are the physiochemical parameters of ointment as well as oral dose: color, pH, total soluble solids, specificity, total minerals, conductivity, and lambda max of the extract. Our product is free from heavy metals, prime requirement of a herbal product. And then we did detailed toxicological evaluation in-house, because we have L1 house with us. And we have validated these toxicological studies at renowned toxicological research center, IIT, Indian Institute of Toxicological Research, for acute toxicity, subacute dermal toxicity and oral toxicity in rat and mice, subchronic toxicity, chronic toxicity in rat and mice, mucous membrane irritation test, skin sensitization test in guinea pigs. The LD50 of the product was found more than 2 grams per kg body weight, and dermal toxicity was found 1 gram per kg body weight. In 6 days exposure, no mortality and morbidity was found. No morphological and pathological lesions on any vital organs or tissue. Body weight ratio for organs, no variation. There was no change in the hematological and serological parameter of the animals. We have conducted animal studies from Indian Institute of Chemical Biology to see the efficacy of the product. The 4 group of animals were made. Both sides of the animals were clipped off with the scissor, and one side ointment was applied, and other side the base petroleum jelly was applied. In second group of animals, both sides of animals were clipped off. In one side, ointment was applied. Simultaneously, all animals were fed with the oral syrup. In third group of animals, the animals were clipped off before without ointment application, but fed with the oral dose. And in fourth group of animals, it was a control. No ointment as well as oral dose was given. In first group of animals, in which only ointment was applied on one side, pigmentation started after 45 days, and after 58 days, distinct pigmentation was there in comparison to the control. In the group in which we gave ointment, we applied ointment and gave oral syrup. The pigmentation started after 39 days, And after 49 days, distinct pigmentation was there. And even pigmentation was appeared on the side in which we had not applied the ointment. It means oral dose is also effective. This is the histology, longitudinal section. When we compare the histology of control as well as treated animals, there was significant increase in number and size of hair follicles, because root of the hair follicle is the site of melanogenesis. This shows product is working. Again, similarly, cross-sectional view, there is same result, significant increase in number and size of melanizing center or hair follicles. Cytotoxic studies, MTTF assay was also done in ointment as well as oral syrup, and product was found free from heavy metals. Product was cytotoxicity. There was no such cytotoxicity in the product. These are the clinical trial results. If a girl suffers from the leukoderma, her whole career comes under stake. These clinical trial we have conducted from Government Ayurvedic College and Hospital, Lucknow. These are the photographs of before treatment, after 270 days, before treatment 240 days, and before treatment 240 days, and before treatment 350 days. Again, these are some pictorial evidences. Result of clinical trials. Clinical trial were conducted from Government Ayurvedic College and Hospital, Lucknow. 51% recovered 75 to 100%. 21% showed recovery between 50 to 75%, and 13% recovered 25 to 50% recovery. We also analyzed the blood plasma of leukoderma patients. In almost, we have taken blood of 40% leukoderma patient and compared with the 4 healthy individual. In leukoderma patient, in 50% patient there was no tyrosinase. 60% patient saw lower level of tyrosinase. In 25% patient, though level of tyrosinase was equal to the control or healthy human being, but the copper concentration of copper was very low. In majority of the patient, copper level was very low. In some cases, copper level was equal to control, but tyrosinase enzyme level was drastically low. This is the protocol for use. Ointment has to apply gently on the white spots twice a day, and oral dose given 5 ml diluted with 20 to 25 ml, and for children 25 ml oral dose twice a day with 20 to 25 ml of water. After that, exposure to the sunlight for 10 to 15 minutes. Dietary restrictions are also there. All sour foods should be restricted. Non-vegetarian diet, preserve and fast foods. Dietary suggestion: soaked red gram, enough pulses, green vegetables. Water for consumption should be kept in copper vessel. Advantage: the product, anti-leukoderma herbal product, has no side effect. Being polycompounded, it is more efficacious. It restores immunity and checks new emerging spots and progression of the disease. Cheap and easy mode of therapy. Thank you.
Any queries? Thank you so much, sir. I thank you on behalf of University of Patanjali Research Institute. Your work and technology transfer is extraordinary. I thank you so much, sir. जोरदार करतल ध्वनियों से धन्यवाद ज्ञापित कीजिए डॉक्टर H.K. पांडे जी का। इसी क्रम में आगे बढ़ते हुए मैं मंच पे इनवाइट करती हूँ डॉक्टर पंकज बराली जी को, who is serving as scientist in North East Institute of Science and Technology, और उनका जो टाइटल है और टॉपिक ऑफ डिस्कशन है leveraging traditional knowledge based bio resources for Northeast India and bio prospecting. I welcome you on the dais, sir.
नमस्ते। परम पूज्य Acharya ji, and the delegates present here. I am Dr. Pankaj Bharali, joined as a scientist in CSIR NIST in 2020. Before that, I was there in Arunachal Pradesh. As yesterday Swamiji mentioned about that, we are blessed with the Himalayas. So, I am coming from the easternmost Himalaya of entire Indian Himalayas. We have this western Himalayas coming from Jammu Kashmir to Uttarakhand, Himachal, and eastern Himalayas with existing 8 states with different topography and geography. So, today I am going to deliver one talk on the leveraging traditional knowledge-based bioresources of Northeast India and its bioprospecting. In front of this August gathering, just I want to share my research experience with the young budding students, which will become the future of this country. So, as we know, India is the world biodiversity hub from easternmost part to western Himalaya. So, I will directly go to the eastern Himalayan region. So, dear students, I think you never visited the eastern Himalayan region, especially the hilly station, Arunachal Pradesh. So, it's basically we call as a land of rising sun, where the sun rises first for the country. So, that is the Dong village, where the sun rises for the first in the country. Next, it's huge diverse. Northeast India, not only either a place of rising sun or ethnic and cultural diversity, but also a mega biodiversity center and count as a hotspot. The region occupies 7.7% of India's total geographical area, supporting 50% of the flora, 800 species, of which 31.58% is endemic to this region. This region is totally unexplored. With the traditional diversity, they have their own tradition and ethnic food, they have their own languages. So, based on those things, we are going to the diversity study of these regions. So, here is a statistical demonstration of the biodiversity of Northeast India, starting from orchids, bamboo, medicinal plants, rare and threatened plant species, aquatic flora, butterflies, and all the other species are existing in this region. So, as my topic is bioprospecting, we can divide this bioprospecting into 3 categories: chemical prospecting, gene prospecting, and the bionic prospecting. Under the chemical prospecting, we can find out the different chemical constitutions in the different flora, and under this genes prospecting, as scientists from CSI NIST, we are doing the gene prospecting of the different bioresources of Northeast India. And also with the bionic prospecting, with the demonstration of the citronella and lemongrass processing unit at different parts of Arunachal Pradesh as well as Northeast India. So, based on these resources, I'm going to talk about the wild bioresources of Northeast India. Especially, there are lots of wild fruit species. We have documented around 200 wild fruit species from this region. Especially, we know about the kiwi, but in Arunachal Pradesh, do you believe it or not, in wild, there are 2 different species of wild kiwi which are existing in the district of West Kameng as well as the Anjao district. Coming to the oil yielding plants, in Arunachal Pradesh and Northeast India, we have different wild oil yielding plants which will be a good source for the rural economy. We have the citronella and lemongrass. Now entire CSI labs are working on citronella and lemongrass. But I came across with different wild species of citronella and lemongrass in different parts of the states. So this work is already published in our journal Medicinal Plant. You can enjoy the beauty of the wild bananas in the Eastern Himalayas regions. Those bananas, we have around 12 different wild bananas which are so nutritious, and all parts of these banana species are used by the local people. Another interesting species which is called as a tache by the local people of Arunachal Pradesh. It's called, the scientific name is Wallicia oblongifolia, belongs to the palm family. The people of Arunachal Pradesh use the trunk of this plant as a alternate food supplement of the rice. The people use these plants during the any type of food scarcity situation. They use these plants as a famine food condition because they are during the zoom cultivation as well as the landslides. Those people, they use these species as a famine food. So this plant is used also for the pigs as well as other animals, and we have developed one technology, and we have applied for a patent for the polymers produced from this trunk of these plants. Now come to the another species which is also found in the western Himalayas. This is Elysium graffiti, which we have, these fruits are used as a carminative and aromatic stimulant, and we have profiled as HPLC, and we have found these polyphenols having gallic acid, pyrocatechol, chlorogenic acid, synergic acid, synapic acid, and all. And the GCMS profile proved that there are around 24 different volatile compounds present in the seeds of this, fruits of this species, Elysium graffiti. And traditionally, it is believed that this fruit has the immune boosting property, and we are working on the immune boosting property in the in vitro as well as in vivo experiment. Another interesting species is piper. This is called Himalayan piper. This piper molesua used by the local people as a vegetables, and we are working on this for the immune boosting property, and these polyphenols are already quantified with the UHPLC method. Very interesting plant, this is the Pyra specia, which is used by the local people as an alternate of tea in the high altitude regions of Arunachal Pradesh. These species, they just collect the dry leaves and decoct with the hot water, and they use in the morning time, and they believe that this species boost the immunity for the people. Then we have characterized the all biochemical profiling of these three species, and it shows good nutrition components from all the micronutrients as well as macronutrients. And the all in vitro bioactivity assay also we have studied. Then this free radical scavenging activity, superoxide radical scavenging activity, and protein degeneration inhibition activity also performed, and it shows good activity. This is a cell proliferation assay in the THP1 cell with methanol extract of pyrus as well as this Elysium graffiti seed extracts. In vitro cell viability study against CC1 hepatocytes, because it is believed that this plant species has some hepatoprotective activity also. And after the treatment, none of the treatment did not cause any significant change in cell viability by Ulmer blue assay. Also, we have studied the effect of crude extract of pyrus and Elysium graffiti on intracellular triglyceride level, and the treatment with pyrus and Elysium graffiti significantly reduced the intracellular triglyceride level at 10 microgram per ml and 20 microgram per ml against this sodium palmitate. So after that, with the help of this UHPLC analysis data of the polyphenols, we have studied the in silico study against CYP2E1 and SIRT1 proteins with the protein receptor. This molecular docking validated binding of detected polyphenols of Pyra specia against the two target proteins CYP2E1 and SIRT1. These are the docking results of these polyphenols from Pyra specia species. So based on the traditional knowledge, we are going to develop one product, and which will be called as a T-Zen. As the tea board people told that we cannot use the other plants as a tea term, so we can say it's a T-Zen from this species. Now coming to another species, this is Gymnocladus assamicus. I work during my PhD work on this species. It's used as a herbal detergent, or it's an anti-lice agent by the people of Arunachal Pradesh. So we have determined the biosurfactant from this species as well as the mosquito larvicidal activity from this saponin extracted from Gymnocladus assamica. And it shows different types of iturin and iturin homologs from these species. So another one important prospect of these traditional green leafy vegetables, I am talking about the wild species, wild herbs of Northeast India. Acharya, I can convey one message that in our tradition, the Assamese people, during the Bihu time, we use more than 101 vegetables in a single curry. So our forefathers and grandfathers always tell that these vegetables and this type of curry always boost our immunity for entire year. So I am going to establish that beliefs, traditional beliefs. So we are working on this 101 different types of vegetables, and we are going to make a mixture of this, and we are studying the proximate composition as well as this mineral contents of these traditional green leafy vegetables. It is more than 135, but out of those 135 species, we are using 101 different vegetables. So yes, dear students, always we are getting some new species. Always in the journal like Phytotaxa, Annals of Botanica, Nordic Journal of Botany, Phytotaxa, we are getting new species discovery. So here is an interesting message that Itanagar times like that, we are getting new species, new species. So my mind always, as a young student, young researcher, I always, as Samiti told, we have to, if we want to know the plants, we have to be there in the field. So I was there in the alpine regions for 20 days continuously without contacting any persons to explore the alpine floristic flora of, to understand the alpine flora of Arunachal Pradesh. And I have collected more than 340 alpine species from Arunachal Pradesh. It was in Tawang as well as Anjao and Anandi district. 20 taxa were recorded as the new to Arunachal India. About 40 additional species previously recorded from Sikkim were possibly new for Arunachal Pradesh. Then Essensua chrysia, it's a very interesting orchid discovered after, it is the second time discovery. From that discovery, we have found some interesting species from the alpine regions. I want to just show the photographs of these species. This is the dioecious species, Belanophora. Then this is Memorias, very interesting flower. Then Sosoria species. So, from this, I am showing here is the distribution of these species of the alpine regions, which are recorded as a new to Arunachal Pradesh, as well as 20 of these are new to India. This is one very interesting Certia species. In Ayurveda, it is there that Certia seriata. But this species was not recorded from Arunachal Pradesh, and the people has a traditional knowledge that this species is used as an anti-diabetic agent. Another interesting species. Dear students, you can enjoy this beauty. You can find out two difference between these two species. See, there are in the bulb position there is a difference, and ultimate after examination we come out that this is a new species to science. This is Rhodiola sedoides from Tawang. This is also one interesting genera, Gentiana. From NBRI people are working, but I can say that if we can do some work on the anti-diabetic property of this Gentiana species, I can assure that lots of products will come from this species. So it is pedicularis, then again this is pedicularis baticularis, and again interesting balsam from Arunachal Pradesh. This asymmetric impatient species, and after examination we came that this one also was a new species to science. This is Impatiens pyrorhizia due to the yellow color of the roots, so we gave the name as a pyrorhizia. This, I talked with Bhatnagar sir yesterday about the orchids. This is an interesting orchid, Essensua chrysia. It is second time described by us from Arunachal Pradesh. These are alpine orchids. You can see the beauty of the hedychium in the eastern Himalayan regions. These are lots of diversities are there. So this is Monotropism, Azientia. Yes, this is the world's largest orchid reported from Arunachal Pradesh. Galeola. It's a saprophytic orchid. But to do the work in the eastern Himalaya is not an easy task. Can you recognize one small hut? We were there for 20 days in that small hut, showing in this picture. So As here written, rule number 1, keep your boots always dirty. There is a risk. It's not easy to do the field survey in Anjao district as well as entire Arunachal Pradesh. We have to face lots of difficulties. These are the situations when I was there during my field study. So I talked with Acharya that we have to do lots of things for the Eastern Himalayas. There is a location I am showing. This is the easternmost part of Arunachal Pradesh, which is a tri-junction of China, Myanmar, and India. None of the scientists were there in that particular location. If somebody from the young students can go there, I can assure you that you can discover lots of species, and that will be new to science. So now, a last question to the students. What is this? Can you give one answer? Pardon? It's a pipe, right? But being a taxonomist or being a scientist, you cannot say that it's a pipe. This is a picture of the pipe, right? So we have to examine ourselves the plants that it is not a pipe, that it's a picture of a pipe. Am I right? So that curiosity should be there to study the taxonomy and the plant science. So yes, but it's so difficult to ask the local people to conserve the bioresources of Northeast India because they have such traditions. We cannot ask them to stop killing the animals. I can show these pictures, sir. You see, they have a tradition to offer the animals, wild animals to their sisters on one particular day. So how can we stop this type of culture? So one side tradition, one side culture, one side conservation. So it should be really serious issues. I have taken these pictures. Within a single family, this mass of wild animals were hunted and they have consumed. So this is our duty to conserve our nature. Yes, in the alpine region, this yak, it is also going to be extinct one day. So my acknowledgement to Rao sir, because I don't know anyone from Patanjali Research Institute. So Rao sir, thank you so much, and Acharya sir, and my director, he allowed me to present here, and yes, all the ethnic people from Arunachal Pradesh as well as Northeast India. Thank you very much.
Thank you so much, sir. Any queries from the audience? So thank you so much.
Thank you.
I thank you on the behalf of organizing committee and Patanjali family. Thank you so much. अब है समय धन्यवाद ज्ञापित करने का, तो मैं हमारे एमिनेंट स्पीकर्स जिन्होंने अपना इतना प्रीशियस समय हमें दिया, इतना कीमती समय निकाल कर वो इतनी दूर दूर से आए, हम उनका हार्दिक धन्यवाद करते हैं। और इसके साथ साथ हम धन्यवाद करते हैं हमारी ऑडियंस का जिन्होंने इतने अच्छे ढंग से इसको सुना, हमारे स्टूडेंट्स का। और एक बार सभी बच्चे पूरे अपने लिए क्लैप करेंगे क्योंकि बहुत ही अच्छे से डिसिप्लिन मेंटेन करते हुए रहे आप। और मुझे लगता है ये एक नॉलेज एम्पावरिंग सेशन आपके लिए रहा क्योंकि विभिन्न विषयों को इसमें कवर किया गया। तो अब मैं पूज्य आचार्य श्री से अनुरोध करूंगी, कृपया वो मंच पे आगे आएं और हमारे जितने भी अभी गेस्ट स्पीकर्स आए थे उनको एक मोमेंटो देकर उनको अलंकृत करें। सबसे पहले मैं आदरणीय डॉक्टर UN दास जी जो हमारे फर्स्ट की नोट स्पीकर हैं, मैं उनसे आग्रह करूंगी कृपया वो मंच पे आएं। आपकी तालियां बजती रहनी चाहिए। इतना समय निकालकर, अपना कीमती समय निकालकर इतने एमिनेंट साइंटिस्ट हमारे बीच में हैं। बहुत बहुत धन्यवाद, बहुत बहुत आपका धन्यवाद, आभार। मैं अनुरोध करूंगी डॉक्टर राखी चतुर्वेदी जी से, कृपया करके वो मंच पे आगे आएं। हम लोग बहुत धन्यवाद ज्ञापित करते हैं मैम, आप इतना समय निकालकर IIT गुवाहाटी से हमारे बीच आई। मैं डॉक्टर के राजामणि जी से अनुरोध करूंगी कृपया वो मंच पे आगे आएं। ये हमारा समर्पण है, ये हमारा विश्वास है, हमारी श्रद्धा है आपके लिए। बहुत बहुत धन्यवाद आपका। मैं अनुरोध करूंगी डॉक्टर टी शेखर जी से। तालियां रुकनी नहीं चाहिए, हम लोगों के लिए गौरव का क्षण है। बहुत धन्यवाद सर। मैं अनुरोध करूंगी डॉक्टर संगीता सक्सेना जी से। बहुत कम समय में ही बहुत अच्छा उद्बोधन इन्होंने दिया और बहुत अच्छे अपने विचार हमारे बीच रखे। बहुत बहुत धन्यवाद मैम। मैं अनुरोध करूंगी डॉक्टर हेमंत पांडे जी से। इनका आशीर्वाद, इनका मार्गदर्शन पहले भी हमें मिलता रहा है। बहुत बहुत धन्यवाद सर। और सभी वरिष्ठों में से हमारे यंग एनर्जेटिक साइंटिस्ट डॉक्टर पंकज जी से मैं अनुरोध करूंगी, कृपया वो मंच पे आगे आए। आपका हम हृदय की गहराइयों से धन्यवाद ज्ञापित करते हैं। हमारे बीच दो और साइंटिस्ट हैं, डिस्टिंग्विश्ड गेस्ट हैं। मैं अनुरोध करूंगी डॉक्टर अर्चना जी से, कृपया वो मंच पे आगे आएं। और पूज्य आचार्य श्री से अनुरोध करूंगी कि उन्हें एक मोमेंटो देकर अलंकृत करें। डॉक्टर अर्चना जी। इसके साथ मैं अनुरोध करूंगी हमारे डिस्टिंग्विश्ड गेस्ट डॉक्टर अनिल कुमार ठाकुर जी से, कृपया वो मंच पे आगे आएं और आशीर्वाद प्राप्त करें आचार्य श्री का। बहुत बहुत धन्यवाद आप सबका। इसके साथ ही क्योंकि कल ओरल प्रेजेंटेशंस हुए थे, पोस्टर प्रेजेंटेशन कंपटीशन हुआ था, उसके रिजल्ट्स भी आ चुके हैं। सबसे पहले मैं पोस्टर प्रेजेंटेशन के विनर्स को मैं बुलाना चाहूँगी मंच पे, और इसके लिए मैं धन्यवाद करना चाहूँगी हमारे जजेस का भी, जिन्होंने एक अनबायस्ड वे में बहुत ही अच्छा और अंकों के आधार पर एक एक कंपोनेंट को अच्छे से वैलिडेट किया। सो मैं फर्स्ट प्राइज गोज टू डॉक्टर रितु पालीवाल and her title was Thyrogrit, a polyherbal formulation restores thyroid function in rat. डॉक्टर रितु पालीवाल, मैं आपको मंच पे आमंत्रित करती हूँ। She is scientist in Drug Discovery and Development Division, पतंजलि रिसर्च इंस्टीट्यूट। तालियां रुकनी नहीं चाहिए। इन यंग बडिंग साइंटिस्ट ने ऐसा इंपॉर्टेंट कार्य जो है वो प्रस्तुत किया और जजेस ने, ज्यूरीज ने इनको हाईएस्ट मार्क्स दिए। डॉक्टर रितु पालीवाल जी। सेकंड प्राइज गोज टू डॉक्टर जितेंद्र कुमार। हाउएवर ही इज नॉट फिजिकली एट प्रेजेंट क्योंकि उनको किसी कार्य के लिए जाना पड़ा, बट आई अनाउंस his topic, zinc oxide nanomaterials, cytotoxicity and their remediation by bioactive molecule viteferin A. Third prize goes to Mayuri Karnal, NDRI Karnal. Third prize goes to Mayuri Shri Krishna Kale, and her topic is enhanced antipsoriatic activity of psorylene through niosomal gel formulation, and she is also from Lucknow. Mayuri ji, please come fast. इसके साथ साथ ही मैं अनुरोध करूंगी ओरल प्रेजेंटेशन के विनर्स के लिए भी। So first prize goes to Ina Chaudhary ji, Gurukul Kangdi University. Ina Chaudhary ji. Second prize goes to Dr. Ashish Bharti ji, Patanjali Ayurved College के Dr. Ashish Bharti ji. Third prize goes to जीत राम चौधरी जी। Third prize goes to Dr. Jeet Ram Chaudhary ji. I invite you on stage. ये हम सब के लिए एक मेमोरेबल इवेंट है, तालियां रुकनी नहीं चाहिए। इन सब के पुरुषार्थ के लिए, इन सब के पराक्रम के लिए, आप लोगों को इनका उत्साहवर्धन के लिए, संवर्धन के लिए तालियां बजाते रहिए। आपकी तालियों की आवाज कम नहीं पड़नी चाहिए। डॉक्टर जीत राम चौधरी जी। बहुत बहुत धन्यवाद पूज्य आचार्य जी। मैं सबसे आग्रह करूंगी कि वो मंच पर आप विराजमान होइए। नहीं, 2 मिनट। अब क्षण है, क्योंकि हम कंक्लूड कर रहे हैं और 2 दिन का ये सत्र जो है, फर्स्ट डे तो हमारा रहा एग्रीकल्चर का और सेकंड डे हमारा रहा हर्बल रिसर्च का। तो सबसे पहले तो मैं कुछ शब्द जो है वो थैंक्स के बोलना चाहूँगी। सबसे पहले पूज्य स्वामी जी महाराज, जिनका एक उनकी ऊर्जा का प्रताप आप सब यहाँ देख रहे हैं, आप सब ने महसूस भी किया है। हम लोगों ने फीडबैक्स फॉर्म फिल करवाए थे क्योंकि हम जानना चाहते थे कहाँ हमारी त्रुटियां रह गई, कहाँ पर हम और इसको इम्प्रूव कर सकते हैं। तो मुझे बहुत खुशी है ये बताते हुए कि आप लोगों ने बहुत अच्छे और सेटिस्फैक्ट्री रिस्पांस दिए हैं। फिर भी यदि हमारी कोई त्रुटि रह गई हो, कहीं कुछ कमी रह गई हो, तो भविष्य में हम उसको जरूर पूर्ण करने का प्रयास करेंगे। इसी के साथ मैं धन्यवाद करना चाहूँगी हमारे जो सोसाइटी के मेंबर्स हैं, हमारे जो NMPB नाबार्ड के स्पॉन्सर्स हैं, उसके साथ साथ यूनिवर्सिटी ऑफ पतंजलि का स्टाफ, उनके स्टूडेंट्स का भी, जिन्होंने बहुत अच्छे से ये मेंटेन किया, एक डेकोरम मेंटेन किया, यूनिवर्सिटी कॉलेज के स्टूडेंट्स का भी, और उसके साथ साथ PRI की पूरी टीम का, जिन्होंने दिन रात अथक परिश्रम करके इस इवेंट को बहुत अच्छे से ऑर्गेनाइज किया। सबसे उसके अतिरिक्त मैं एक बड़ा इम्पोर्टेंट विषय था, क्योंकि गुरु कभी भी अपने लिए कोई अगर कोई क्रेडिट की बात आती है तो कभी भी अपने लिए कोई क्रेडिट अपने पास नहीं रखते हैं, और ये संत की महिमा भी होती है। तो मैं 2 शब्द पूज्य आचार्य श्री के लिए पूरी PRI टीम की तरफ से आप लोगों के सामने रख रही हूँ। उसके बाद एक छोटा सा एक समर्पण का हमारा एक मंत्रोच्चार है जो कि हमारी टीम ने तैयार किया है, और उसके पश्चात जो है हम आगे के कार्यक्रम की तरफ बढ़ेंगे। पूज्य आचार्य श्री का जो जीवन का एक पूरा हम लोगों ने देखा, पुरुषार्थ और पराकाष्ठा युक्त वह जीवन रहा। मैं बहुत कम शब्दों में केवल ये बात बोलना चाहूँगी, क्योंकि शब्द इसके लिए कम पड़ जाएंगे, कि कभी तो उनमें एक बहुत बड़े IT प्रोफेशनल का एक हम प्रतिबिंब देखते हैं कि बहुत बड़े IT प्रोफेशनल हैं। कई बार वेदांग पूरे उपनिषद विद्वान दिखते हैं कि उसमें जितने भी अमूल्य ग्रंथ हैं, सौमित्र संहिता हो गई, और जितने भी हमारे आर्ष ग्रंथ हैं, कि उनके बहुत ही विद्वान पंडित दिखते हैं। उसके अलावा कभी कभी परम अनुभवी चिकित्सक दिखते हैं, कभी बहुत ही ज़्यादा कठोर अनुशासक दिखते हैं। इस प्रकार से दिखते हैं जैसे पिता डांटते हैं तो उसी प्रकार से सबको अनुभव होता है, लेकिन कभी कभी इतनी दयालुता से इतने परम सुधारक दिखते हैं। तो विभिन्न पूज्य आचार्य श्री के रूपों का मुझे ऐसा लगता है इस भवन में बैठे सभी हमारे बच्चों ने, हमारे स्टाफ ने, फैकल्टी मेंबर्स ने उनका ये दर्शन जरूर किया है। आज इस मंच से ये पूरा का पूरा इंसाइक्लोपीडिया ग्रंथ, पूरा का पूरा वर्ल्ड बोटेनिकल डिक्शनरी ग्रंथ, और उसके अलावा जितने भी ग्रंथ हैं, आज हम सब मिलकर समर्पयामी समर्पयामी अत्र भवद्भ्य समर्पयामी, हम आपको समर्पण करते हैं। आपकी ही ऊर्जा है, आपकी ही ऊर्जा है जो हम सब में संचारित होती है, जिसका प्रस्फुटन हम सब में होता रहता है। कभी कभी उसका प्रस्फुटन होकर कुछ अच्छे कार्य कर लेते हैं, कभी कभी कुछ न्यूनता रह जाती है, लेकिन एक ही लक्ष्य है, एक ही मंतव्य है कि इसी दिशा से आगे बढ़ते रहेंगे। समर्थ गुरु की शरण में आ चुके हैं, इसी तरह आगे बढ़ते रहेंगे। इसी शब्दों के साथ मैं आमंत्रित करती हूँ मंच पे करुणा जी और स्वाति जी को, जो कि समर्पणम का एक मंत्र की जो एक मंत्र है वो आपके सामने प्रस्तुत करेंगे। और इसके साथ आमंत्रित करती हूँ पूरी PRI की टीम को कि स्टेज पर आकर पूज्य आचार्य श्री से आशीर्वाद ले। ओम।
शरीरं सुरूपं तथा वाक्कलत्रं यशश्चारुचित्रं घनं मेरुतुल्यं मनश्चेन्न लग्नं गुरोरङ्घ्रिपत्रे ततः किं षडङ्गादिवेदो मुखे शास्त्रवेद्या कवित्वादिगद्यं सुपद्यं करोति मनश्चेन्न लग्नं गुरोरङ्घ्रिपत्रे ततः किं विदेशेषु मान्यः स्वदेशेषु धन्यः सदाचारवृत्तेषु मत्तो न चान्यः मनश्चेन्न लग्नं गुरोरङ्घ्रिपत्रे ततः किं क्षमामण्डले भूपभूपालविन्दै सदा सेवितं यस्य पादारविन्दं मनश्चेन्न लग्नं गुरोरङ्घ्रिपत्रे ततः किं यशो मे गतं दिक्षु दानप्रताप जगद्वस्तु सर्वं करे सत्प्रसादा मनश्चेन्न लग्नं गुरोरङ्घ्रिपत्रे ततः किं कलत्रं धनं पुत्रपौत्राणि सर्वं गृहं बान्धवा सर्वमेतद्विजातं मनश्चेन्न लग्नं गुरोरङ्घ्रिपत्रे ततः किं षडङ्गादिवेदो मुखे शास्त्रवेद्या कवित्वादिगद्यं सुपद्यं करोति मनश्चेन्न लग्नं गुरोरङ्घ्रिपत्रे ततः किं विदेशेषु मान्यः स्वदेशेषु धन्यः सदाचारवृत्तेषु मत्तो न चान्यः मनश्चेन्न लग्नं गुरोरङ्घ्रिपत्रे शरीरं सुरूपं तथा वाक्कलत्रं यशश्चारुचित्रं धनं मेरुतुल्यं मनश्चेन लग्नं गुरोरङ्घ्रिपत्नी ततः किं षडङ्गादिवेदो मुखे शास्त्रवेद्या कवित्वादिगद्यं सुपद्यं करोति मनश्चेन लग्नं गुरोरङ्घ्रिपत्नी ततः किं विदेशेषु मान्यः स्वदेशेषु धन्यः सदाचारवृत्तेषु मत्तो न चान्यः मनश्चेन लग्नं गुरोरङ्घ्रिपत्नी ततः किं क्षमामण्डले भूपभूपालवृन्दै सदा सेवितं यस्य पादारविन्दं मनश्चेन लग्नं गुरोरङ्घ्रिपत्नी ततः किं
बहुत बहुत धन्यवाद। ये पूरी की पूरी साइंटिस्ट की टीम जो आपको दिख रही है, जिन्होंने पूज्य आचार्य श्री के मार्गदर्शन में अथक परिश्रम किया और दिन रात विभिन्न प्रकार की व्यवस्थाओं में ये लोग लगे हुए थे। आप लोग की जोरदार तालियों से इन लोगों का अभिनंदन कीजिए। ओम। अब एक भव्य सांस्कृतिक कार्यक्रम है।
ओम। अभी सांस्कृतिक कार्यक्रम होना है। सभी जो बच्चे और सभी हमारे वरिष्ठ हमारे संस्थान के हमारे सहयोगी अभिभावक सभी आप यहाँ पर हैं। आप सबसे निवेदन है आप अभी आनंद लेना, बैठे रहना, क्योंकि यह कार्यक्रम का एक सत्र एक तरह से पूरा हुआ है। जो वनस्पति वाला कल क्लिनिकल रिसर्च का पार्ट होना है, कल वो तगड़े से साइंटिस्ट जी दिखाई देंगे आपको, हमारे अनुराग जी, हमारे सभी तगड़े हैं, अच्छा है। और मुझे अच्छा लगता है, मैं अपने बच्चों से भी बाकी लोगों से भी जितने भी हैं, कोई प्रशंसा की बात नहीं है। जिस तरह से यूं तो पतंजलि योगपीठ संस्थान में हर एक व्यक्ति, हर एक बच्चा एक मिशन, एक लक्ष्य को लेकर के यहाँ पर है। चाहे वो पढ़ने वाला बच्चा हो, चाहे वह उनके गुरुजन हो, चाहे वह किसी भी तरह की कोई सेवा में नियुक्त व्यक्ति हो, सभी का पुरुषार्थ, सभी का कंट्रीब्यूशन, सभी की भावनाएं अद्भुत हैं, अविस्मरणीय हैं, अतुलनीय हैं और हमको गौरवान्वित करने वाली है और पूरी देश और दुनिया के लिए एक उदाहरण है। सब अच्छे हैं, पर उन अच्छों में कुछ अग्रणी भूमिका के तहत हम कहीं शिथिल न पड़ जाएं, प्रेरणा भी देते हैं। तो उनमें से अभी तो हम कल भी हैं, परसों भी सब साथ में मिलेंगे पुनः, क्योंकि अभी सब बहनें, सब भाई जो यहाँ पर अभी इकट्ठे होकर के गए थे, अलग तरह का, या तो ऐसा भी हो सकता है कि मुझे इन लोगों के साथ ज्यादा जूझना पड़ता है या ज्यादा संपर्क में रहता हूं, जिस तरह से इन लोगों ने काम किया है। क्योंकि आप सब जानते हैं कि क्योंकि जितने भी यहाँ पर हैं, ये देश की विभूतियां हैं, अपने अपने जगह के सब शंकराचार्य हैं, ये कोई ऐसा भी कम नहीं। तो सब जानते हैं, ये जितने भी प्रोफेसर हैं, कईयों के 10, 20, 30 किताबें भी हैं इनमें से, मुझे पता है। ये बिल्कुल प्रामाणिक बात है। आप लोगों में से भी कई हमारे हैं लिखने में, पढ़ने में बहुत लोग। एक पुस्तक लिखना, क्योंकि देखिए, क्षमा करिएगा, मजाक मनोरंजन में को भाषण तो वही हर रोज दिया जा सकता है। लिखने के लिए यदि लिखना हो तो एक ही चीज को हर रोज नहीं लिखा जा सकता। थोड़ा से शब्दों का, थोड़ा उदाहरण के परिवर्तन करने से वो चीजें चलती हैं, पर लिखने में नहीं चल सकता। तो ये इतना जटिल कार्य होता है, और एक पुस्तक तैयार होना और उस पुस्तक को प्रकाशित करना, ये उससे भी जटिल कार्य होता है। लोग कहते हैं ना, बोले बीपी हाई, बोले या तो लिखने में हो जाता है, नहीं तो बोले जो छपवाने में तो होता ही होता है। और पब्लिशर से तो कोई मिल भी जाता है। विद्या का, विद्वानों का मैंने देखा जो सम्मान होना चाहिए भारत में, तो कम से कम मैं जानता हूँ 2-4 पब्लिशर्स को, वो ऑथर बुक 1 कॉपी दे देंगे, 2 दे देंगे, 3 दे देंगे, बोले हाँ भाई साहब आपकी छाप दी, बोले जाओ। तो ये हालत है। तो उसमें जो टीम आपको दिखाई दे रही थी, मैक्सिमम लगभग 40 से ज्यादा तो इनमें पीएचडी हैं, और बाकी सब पीजी तो लगभग सबने किया ही हुआ है। बाकी जो टेक्निकल टीम थी, जो डिजाइनर थे, टाइपिस्ट थे, सब लोग ये थे। तो दुनिया में आप सबके लिए ये बात इसलिए गौरव वाली बात है, क्योंकि हमारा लक्ष्य है, हमारे अभी 58 वॉल्यूम्स अभी हमारे छपने बाकी हैं। ये 51 हुए हैं, 58 और बाकी है। परंतु 58 का भी जो काम है, कोई 70%, कोई 80%, कोई 50%, कुछ वॉल्यूम्स के काम थोड़े से कम हुए हैं। तो लगभग एक सीमा तक कार्य हो चुका हुआ है। वह अब उस काम को आगे बढ़ा रहे हैं ये लोग, और हमारा लक्ष्य है कि यह 5 जनवरी हमारी इस पतंजलि योग पीठ जो संस्था है, आप सबको पता भी होना चाहिए, 1995 5 जनवरी को इसकी दिव्य योग मंदिर ट्रस्ट के रूप में पहला हमने संस्था का रजिस्ट्रेशन ट्रस्ट का करवाया था। उसके स्थापना दिवस के रूप में 95 से आप लगाइए, अभी 22 हम अभी चल रहे हैं, तो 23 होगा जनवरी में। तो कितने वर्ष हो गए? आपका लगभग 28 वर्ष पूरे हो जाएंगे। तो संस्थान के, और मजेदार बात अभी मैं थोड़ा सा स्मरण आपको करा दूं बच्चों के लिए भी, आयुष मंत्रालय की स्थापना भी 95 को ही हुई थी, भारत सरकार की। तो अब वो मैं तुलना में नहीं चाहूँगा, मैं इतना ही कहना चाहूँगा कि हमने श्रद्धेय स्वामी जी के नेतृत्व में, श्रद्धेय स्वामी जी के आशीर्वाद से, उनके प्रेरणा से कभी भी किसी कार्य के लिए कभी एक बार भी उन्होंने नहीं कहा कि क्यों कर रहे हो, क्या करने वाले हो, क्या करते जा रहे हो। जो हमको समझ में आया श्रद्धेय स्वामी जी का, उसके लिए हमेशा समर्थन रहा। इसीलिए यह कार्य इस रूप में, क्योंकि इस कार्य को करते समय मैं जब इन सारे बच्चों को देख रहा था, ये इकट्ठे ऐसे नहीं हुए हैं, एक एक व्यक्ति को करके हमने इकट्ठा किया। शुरू में 2 आए, 4 आए, 6 आए, 10 आए, कुछ गए भी, संसार की व्यवस्था है, पर जो जमे रहे वो ऐसे जमे रहे, और अभी भी इस तरह से ये लोग जमे हुए हैं। इनको कह दें अभी जैसे दिन भर काम किया हुआ, अब कहीं नहीं जाना, अभी 3 दिन यहीं रहो, ये नहीं पूछे ये कि क्यों रहे। इस तरह से ये लोग काम करते हैं। यानी इन्होंने कभी कर्मचारी जो शब्द है ना, यदि कर्मचारी इनको कह दिया जाए तो बेमानी है। ये एक मिशनरी हम कह सकते हैं उनको, या एक कर्मयोगी कह सकते हैं। और उनमें भी बहनों की टीम ज्यादा है, भाइयों से ज्यादा। ये बहनें हमारी बैठी हैं, बहनों की ज्यादा समस्याएं होती हैं, क्योंकि घर वाले अलग तरह के समस्याएं खड़ी करे रखते हैं। घर भी संभालो, ऑफिस भी संभालो, इधर भी। पुरुष तो निकल गया तो घर की चिंता नहीं रखता, सोचता है वो तो संभाल ही रही होगी। पर बहन निकलती है तो यहाँ बैठे बैठे भी घर याद आ रही है उनको, वहाँ क्या हो रहा है घर पे। तो ये स्थिति रहती है। तो एक बड़ा उदाहरण, और मैं इस बात को इसलिए कह रहा हूँ, जनवरी दूर नहीं है, और पूरी दुनिया के लिए ब्रिटिश फार्माकोपिया शायद एक रूप में वो बड़ा ग्रंथ माना जाता है। और इसके अतिरिक्त कोई बता रहा था कि अभी नेट पे कोई दूसरा ग्रंथ का भी है कि जो 100 वॉल्यूम से भी ज्यादा है। अभी मैं चेक करा लूंगा उसके पेजेस, टोटल कंटेंट कितना है। हमको तो ये लगता है कि जब हमारे 109 वॉल्यूम्स कंप्लीट हो जाएंगे, ये दुनिया का अपने आप में एक विषय पर, एक सब्जेक्ट को, एक चीज को लेकर के लिखा गया दुनिया का सबसे बड़ा ग्रंथ बनेगा। और दूसरी बात ये, हमारे आयुर्वेद वाले बच्चों, आप लोगों के लिए बड़ी बात है, यह सब जानते हैं कि ये ऐसा ऐतिहासिक काम है, क्योंकि पहले तो हुआ नहीं, आगे करने के लिए हमने छोड़ा नहीं। तो न भूतो न भविष्यति, पहले किसी ने किया नहीं, आगे के लिए क्या करेंगे। हमारे 6 लाख तो रेफरेंसेज हैं, इसके बाहर से तो कुछ लिखा जा सकेगा नहीं। लिखना हो तो लिखेगा क्या आदमी, तो इसलिए दोबारा भी कभी होगा नहीं। तो आप सब जितने यहाँ बैठे हो ना, आप सब इतिहास पुरुष हो गए हो। इस इतिहास की इस घड़ी के साक्षी हैं। कल भी साक्षी थे, आज भी हैं, कल भी रहोगे, परसों भी रहे। इस साक्षी पने को हम मजबूती देने के लिए हमने इस तरह से लगा के रखी, क्योंकि एक बार विमोचन हो गया तो पता भी नहीं चले। आदमी देखते देखते भी सोच लेता है छोटी छोटी तो नहीं है, ये तो बड़ी बड़ी है। एक एक किताब को उठा करके लग कहते ये तो ग्रंथ है। ग्रंथ मतलब काफी लंबा चौड़ा। तो यह कार्य ये हुआ है, आप सबको पता है, और आप सबकी भी भावनाएं हैं। क्योंकि कोई भी अच्छा काम होता है ना, प्रत्यक्ष सहयोगी तो अपने होते ही हैं, परोक्ष न जाने कितने लोगों की शुभकामनाएं हैं, भावनाएं हैं, संवेदनाएं हैं। पूरे देश को पता चल रहा है कि ऐसा काम हो रहा है, तो करोड़ों लोगों को हृदय में प्रसन्नता है, उमंग है, उत्साह है। उससे आप सबको और हमको ऊर्जा मिलती है। तो उसी ऊर्जा से यह काम होता है। तो सभी विद्वज्जनों का जो यहाँ पर पधारे, कुछ कल भी हमारे साथ होंगे, कुछ को जाना होगा। कल नई टीम हमारे बहुत सारे, कल विशेष करके सभी विशिष्ट हैं। आप क्योंकि ज़्यादातर जड़ी बूटियों से संबंधित विषयों में हैं, तो नॉर्मली आयुर्वेद का प्रति प्रेम भाव रहता ही है। पर कल वाले जो आ रहे हैं वो विशेष करके, क्योंकि वो एलोपैथिक फील्ड के धुरंधर लोग हैं, तो प्रायः हम ऐसा मानते हैं कि अभिरुचि वो आयुर्वेद की बात करेंगे तो एक और भी अलग लगेगा हमको। तो सब लोगों के लिए पुनः मैं सबको बहुत प्रणाम करता हूँ, आप सबको आपके आगमन के लिए। और आप लोग ऊर्जा के साथ बैठे रहिए। अब इनका कोई कल्चर प्रोग्राम होगा। और पुनः विक्टोरिया जी, आपकी टीम और सबको बहुत बहुत शुभकामनाएं बच्चों। कल भी दूंगा, परसों भी दूंगा। ठीक है, धन्यवाद। हाँ हाँ, इनके साथ आप बीच में चलो। ये इनका ये जो वेद जी भी आई है। आइए डॉक्टर साहब, आप लाइन में लोगो ना, आप आओ ना, आप पीछे क्यों? आप
बहुत बहुत धन्यवाद पूज्य आचार्य जी। आपकी ऊर्जा, आपका मार्गदर्शन ही है जो हम सब में, पतंजलि के पूरे परिवार में संचरित होता रहता है। मैं निवेदन करूंगी सभी हमारे आगंतुक महानुभावों से कि कृपया करके वो आगे मंच पर बैठें। एक बहुत ही सुंदर कार्यक्रम, हमारे पतंजलि की एक पूरी झलक आज आपके सामने आने वाली है। हमारे छोटे छोटे बच्चे, हमारी बेटियां, हमारे बच्चे किस प्रकार से पतंजलि की पूरी झलक आपके सामने आज प्रस्तुत करने जा रहे हैं। मैं मंच पे आमंत्रित करती हूँ आगे के मंच संचालन के लिए आदरणीय स्वामी परमार्थ देव जी को। बहुत ही आदर के साथ जोरदार करतल ध्वनियों के साथ स्वागत कीजिए स्वामी परमार्थ देव जी महाराज का।
बहुत बहुत धन्यवाद आदरणीय डॉक्टर वेद प्रिया जी का। एक बार जोरदार करतल ध्वनि करेंगे और उत्साह के साथ परम पूज्य आचार्य श्री जी महाराज, परम पूज्य स्वामी जी महाराज का पावन मार्गदर्शन, उनकी प्रेरणा, पतंजलि विश्वविद्यालय के हमारे विद्यार्थी सबसे पहले प्रस्तुति लेकर आ रहे हैं। समूह गायन के लिए मैं आमंत्रित करूंगा बहनों को। गुणवान बने, यशवान बने, क्योंकि गुणों से ही व्यक्ति की पूजा होती है। उसके गुण, कर्म, स्वभाव ही उसको श्रेष्ठता प्रदान करते हैं। तो एक बार करतल ध्वनि के साथ पुनः पतंजलि विश्वविद्यालय की बहनों को आमंत्रित करता हूं समूह गान के लिए। क्षमा दमोस्तेयम शौचम इंद्रिय निग्रह धीर विद्या सत्यम क्रोध दशकम धर्म लक्षणम। धीर वीर गंभीर हम बने, इस माँ भारती की सच्ची आरती अपने कर्मों से उतारें, ऐसा यह भाव सुंदर गीत के माध्यम से आपने सुना। इन सभी छात्राओं के लिए एक बार पुनः उत्साह के साथ करतल ध्वनि करेंगे। और अब आपके समक्ष मैं आमंत्रित करता हूँ सांस्कृतिक नृत्य के लिए हमारे एमएससी की विद्यार्थियों को, छात्राओं को। गढ़वाली, क्योंकि हिमाचल और उत्तराखंड की एक परंपरा जो सबको आकर्षित करती है, ये परंपरा से जुड़े हुए जो नृत्य हैं, जो कलाएं हैं, वास्तव में वो हमारे विनम्रता का, हमारी संस्कृति का प्रदर्शन करते हैं। तो आपकी जोरदार करतल ध्वनि के बीच में गढ़वाली नृत्य के लिए छात्रा अर्पिता जी को, शिखा जी को आमंत्रित करता हूँ। लोकगीत, लोक संगीत, लोक नृत्य उत्साह से, उमंग से, प्रसन्नता से हमको सराबोर कर देते हैं और यही भारतवर्ष की निशानी है। यही हमारे परंपरागत जो कार्यक्रम हैं, जो विरासत है, वो पूरी दुनिया में, पूरे विश्व में उसका एक बहुत बड़ा महत्व है। मैं आमंत्रित करता हूँ एक शौर्य, वीरता से परिपूर्ण एक प्रस्तुति। विजयी भव छात्रा मीनाक्षी जी को, पतंजलि विश्वविद्यालय की छात्रा हैं, एक बार जोरदार करतल ध्वनि करेंगे।
तिनका था हमने सवारा, अपनी वो माटी और घर बारा, लुट रहा ये चमन अपना वतन, आंखों से अपनी लुट रहा ये चमन अपना वतन, आंखों से अपनी। संकल्प बोल के हम तो निकल पड़े, हर द्वार खोल के गगन गए। विजयी भव, विजयी भव, भटकी है रोशनी, है छटपटा रही रोशनी। गगन गए विजय भव, घायल है तेरा जल, तू नदी है राह बदल, पानी बुलबुला रहा है कल कल कल। तू निकल, तू निकल, माटी ने तेरी आज पुकारा, धरती ये पूछे बारंबार, तेरा वतन, आँखों से अपनी लुट रहा ये चमन, तेरा वतन।
विजयी भव। रगों में धीरता वीरता गंभीरता से यह भूमि, यह भारतवर्ष परिपूर्ण है और इस मातृभूमि के लिए हमारे वीर वीरांगनाओं ने अपने प्राणों की आहुति हंसते हंसते दी है। आइए एक योग गीत की प्रस्तुति के लिए मैं आमंत्रित करता हूं हमारी बहुत ही यशस्विनी ब्रह्मवादिनी बहनों को। पतंजलि संन्यास आश्रम की हमारी ये बहनें परम पूज्य श्री स्वामी जी महाराज, आचार्य श्री जी महाराज के पावन आशीर्वाद से शास्त्रों को पढ़ती हैं और शास्त्रोक्त जीवन जीती हैं। शुद्धता, पवित्रता, धीरता, वीरता, गंभीरता से युक्त ऐसी ब्रह्मवादिनी बहनों के लिए एक बार जोरदार करतल ध्वनि करेंगे। सारा योगी बने संसार, कोई न रहे बीमार। हम योगी बने, उपयोगी बने, सबके सहयोगी बने, यही हमारे गुरुदेव का आदेश, उपदेश, निर्देश है। पुनः एक बार जोरदार करतल ध्वनि करेंगे।
सारा योगी बने संसार, सारा योगी बने संसार, जगत में कोई न हो बीमार, जीवन सारा तर जाएगा, जीवन सारा तर जाएगा। सारा योगी जगत में योग का हो विस्तार, जीवन सारा तर जाएगा, जीवन सारा तर जाएगा। सारा योगी बने क्लेशों की गठरी ने मन को था घेरा, आठों घड़ी अब है योग का पहरा। क्लेशों की गठरी ने मन को था घेरा, आठों घड़ी अब है योग का पहरा। मेरा होने लगा उद्धार, अपना, निरोगी काया मन ओम जपना। सम्यक सेवन मन शुद्ध अपना, निरोगी काया मन ओम जपना है योग का यही सार, है योग का यही सार। जगत में कोई ना हो बीमार, जीवन सारा तर जाएगा, जीवन सारा तर जाएगा। सारा योगी बने संसार, जगत में योग का हो विस्तार। जीवन सारा तर जाएगा, जीवन सारा तर जाएगा, जीवन सारा तर जाएगा, जीवन सारा तर जाएगा।
ओम। एक बार पुनः करतल ध्वनियों से इन बहनों का हम हार्दिक स्वागत अभिनंदन करते हैं। सब योगी बने संसार, परम पूज्य परम श्रद्धे योग ऋषि स्वामी जी महाराज, परम श्रद्धे प्रातः स्मरणीय आयुर्वेद शिरोमणि आचार्य श्री जी के पावन आशीर्वाद से पतंजलि योग पीठ के विभिन्न प्रकल्प एक सामान्य से विशेष की यात्रा में आगे बढ़ रहे हैं। हम सभी योग में जीवन जी रहे हैं, तो इसमें केवल इन दो जो महान विभूति हैं इन्हीं का प्रसाद स्वरूप हम सभी योग के माध्यम से अपना जीवन हम संवार रहे हैं। पूरा विश्व वास्तव में आज योग में है, तो वो इन्हीं महान विभूतियों की देन है। अभी जो अग्रिम प्रस्तुति है योग डेमो, पतंजलि विश्वविद्यालय के बालकों द्वारा ये प्रस्तुति तैयार की गई है। वास्तव में प्रत्येक कार्य में योग समाहित है। हमारे प्रातः काल के जागरण से लेकर रात्रि शयन तक प्रत्येक जगह योग विद्यमान है। और ये योग ही जब इसमें लगातार निरंतर श्रद्धा के साथ हम अपने कर्मों में एक निरंतरता श्रद्धा लेके आ जाते हैं, तो वो योग में कार्य हो जाता है। और उस योग का ही एक प्रारूप आप इस योग नृत्य के माध्यम से देखेंगे कि किस प्रकार से शारीरिक स्थिरता, क्योंकि स्थिर सुखम आसनम ऐसा महर्षि पतंजलि ने कहा है, और योग वास्तव में शरीर और मन को स्थिर करता है। वो पूरा समन्वय, शरीर और मन का समन्वय आप इस योग नृत्य के माध्यम से आप देखेंगे। तो इन सभी विद्यार्थियों के लिए एक बार करतल ध्वनि से उनका स्वागत करें, पतंजलि विश्वविद्यालय के बालक।
प्रचंड बोल मस्तकों के झुंड, आज जंग की घड़ी की तुम हार दो। है प्रचंड बोल मस्तकों के झुंड, आज जंग की घड़ी की तुम हार दो। आन बान शान या की जान का हो दान, आज एक धनुष के बाण पे उतार दो। आरंभ है प्रचंड बोल मस्तकों के झुंड, आज जंग की घड़ी की तुम गुहार दो। आन बान शान या के जान का हो दान, आज एक धनुष के बाण पे उतार दो। आरंभ है प्रचंड सो प्राण दे, जो मन करे सो प्राण ले, वही तो एक सर्वशक्तिमान है। मन करे सो प्राण दे, जो मन करे सो प्राण ले, वही तो एक सर्वशक्तिमान है। कृष्ण की पुकार है, ये भागवत का सार है कि युद्ध ही तो वीर का प्रमाण है। कौरवों की भीड़ हो या पांडवों का नीड़ हो, जो लड़ सका है वो ही तो महान है। जीत की हवस नहीं, किसी पे कोई वश नहीं, क्या जिंदगी है ठोकरों पे मार दो। मौत अंत है नहीं तो मौत से भी क्यों डरे, ये जाके आसमान में दहाड़ दो। आरंभ है प्रचंड बोले मस्तकों के झुंड, आज जंग की घड़ी की तुम गुहार दो। आन बान शान या की जान का हो दान, आज एक धनुष के बाण पे उतार दो। आरंभ है प्रचंड। दया का भाव या कि शौर्य का चुनाव, या कि हार का वो घाव, तुम ये सोच लो। वो दया का भाव या कि शौर्य का चुनाव, या कि हार का वो घाव, तुम ये सोच लो। या कि पूरे भाल पर जला रहे विजय का लाल लाल ये गुलाल, तुम ये सोच लो। रंग केसरी हो या मृदंग केसरी हो, या कि केसरी हो ताल, तुम ये सोच लो। जिस कवि की कल्पना में जिंदगी हो प्रेम गीत, उस कवि को आज तुम नकार दो। भीगती मसों में आज फूलती रगों में आज आग की लपट का तुम बघार दो। आरंभ है प्रचंड बोले मस्तकों के झुंड, आज जंग की घड़ी की तुम गुहार दो। आन बान शान या की जान का हो दान, आज एक धनुष के बाण पे उतार दो।
जोरदार करतल ध्वनि, इतना उत्साह उमंग से उल्लास से परिपूर्ण यह प्रस्तुति हमारे पतंजलि विश्वविद्यालय की विद्यार्थियों की। और योग आयुर्वेद ऋषि परंपरा का संवाहक यह प्रतिष्ठान, जो ऋषिकल्प परम पूज्य स्वामी जी महाराज आचार्य श्री जी के पावन सानिध्य में, उनके पावन निर्देशन में चलता है। तो मैं आमंत्रित करूंगा पतंजलि आयुर्वेद महाविद्यालय के विद्यार्थियों को समूह गान के लिए, धन्वंतरी वंदना के लिए। एक बार जोरदार करतल ध्वनि के बीच में हमारे आयुर्वेद महाविद्यालय के विद्यार्थियों को। चरक सुश्रुत धन्वंतरी की यह पावनी परंपरा का निर्वहन करने वाला संस्थान।
कृतं तापत्रया रक्षकं प्राणी प्राणबलप्रदं दिविशदं विज्ञानरत्नाकरं दिव्यं भव्यं ऋषीश्वरं प्रभुवरं धन्वन्तरिं तन्नुमः श्रावं श्रावमहो यदीयसुकथां देवाय देवेष्टपं दर्शं दर्शमहो यदीयरचना ऊर्जायते चाखिलः स्मारं स्मारमुदार दिव्यचरितं दीनोऽपि शूरायते दिव्यं भव्यं ऋषीश्वरं प्रभुवरं धन्वन्तरिं तन्नुमः रोगक्लेशनिशाचरोगविसरो यन्नाम संश्रुत्य वै दैन्यं म्लानिमवाप्य पङ्गुरिवः द्राकान्विषीकायते आयुर्वेददिवाकरं गदतमो हन्तारमर्च्योत्तमं दिव्यं भव्यं ऋषीश्वरं प्रभुवरं धन्वन्तरिं तन्नुमः दुःखापारपयोनिदेहितरणिर् यः पूर्वजैः कीर्तिता आयुर्वेदमहोषधीयुतिततिर् यस्याचलां रक्षति आयुष्यामृतमेव यस्य गदितं भक्त्याः फलं श्रीमदं दिव्यं भव्यं ऋषीश्वरं प्रभुवरं धन्वन्तरिं तन्नुमः कल्याणं निखिलस्य दुःखहरणं सौख्यामृतावर्षणं सल्लक्षं समदर्शि यस्य विमले दिव्यामृते जीवने तस्याशी परिपातु विश्वमखिलं नानामयैः पीडितं दिव्यं भव्यं ऋषीश्वरं प्रभुवरं धन्वन्तरिं तन्नुमः धन्वन्तरिं तन्नुमः धन्वन्तरिं तन्नुमः।
बहुत बहुत धन्यवाद। और आपका उत्साह बता रहा है कि आप बहुत उत्साह में हैं। 10-15-20 मिनट का कार्यक्रम और है। एक बार अपने लिए जोरदार करते ध्वनि करिए। आपके धैर्य को मैं प्रणाम करता हूं। प्रभात 9 बजे से लेकर के अभी तक इस रस का आनंद ले रहे हैं। और संगीत साहित्य, संगीत कला विहीना जो कहा है, तो भारतवर्ष संगीत से, कला से सदा परिपूर्ण रहा है। पतंजलि योग पीठ में भी ऐसे ही हमारे विद्यार्थी जो अनेक कलाओं से युक्त हैं। मैं आमंत्रित करता हूं एक सामूहिक नृत्य के लिए पतंजलि विश्वविद्यालय की बहनों को आपके जोरदार करतल ध्वनि के बीच में।
जल प्रवाह पावितस्थले कलेवलम्ब्य लम्बितां भुजङ्गमण्डमड्डमड्डमन्निनादवड्डमर्वयं चकार चण्डताण्डवं तनोतु नः शिव शिव शिवम् जटाकटाह सम्भ्रम भ्रम निलिम्ब निर्झरी विलोल वीचि वल्लरी विराजमान मूर्धनि ज्वलल्ललाट पट्ट पावके किशोर चन्द्रशेखरे रति प्रशिक्षणम् धराधरेन्द्र नन्दिनी विलास बन्धु बन्धुरः सुरद्दिगन्त सन्तति प्रमोदमान मानसे कृपा कटाक्ष धोरणी निरुद्ध दुर्धरापदि क्वचित् दिगम्बरे मनो विनोद मेतु वस्तुनि ॐ नमः शिवाय। आजा आजा जिंद शामी आने के तले आजा जरी वाले नीले आसमाने के तले जय हो आजा आजा जिंदे शामी आने के तले है, आजा जरी वाले नीले आसमाने के तले है। जय हो जय हो नच नच कोयलों पे रात बिताई है, अखियों की नींद मैंने फूकों से उड़ा दी, दिल दिल तारे मैंने उंगली जलाई है। आजा आजा जिंदे शामी आने के तले, आजा सारी तारे नीले आसमान के तले है। जय हो शुक्र हो जय हो तूने दे जय हो तूने दे जय हो
जोरदार अभिनंदन हमारी छात्राओं का, बहुत सुंदर प्रस्तुति। और अब अगली प्रस्तुति के लिए मैं आमंत्रित करता हूं हमारे विश्वविद्यालय की बहनों को। हमें निज धर्म पर चलना सिखाती गीता रामायण। हमारे भारतवर्ष की परंपरा जो सनातन परंपरा, पुरातन परंपरा है, हम उसके सच्चे संवाहक बने। और यहाँ पतंजलि योगपीठ में आचार्य कुलम, पतंजलि गुरुकुलम और पतंजलि संन्यास आश्रम विश्वविद्यालय के सभी भाई बहन, आयुर्वेद महाविद्यालय के भाई बहन गीता रामायण उपनिषद दर्शन पढ़ करके उसको आत्मसात भी करते हैं। तो इन बहनों के लिए ये गीता रामायण उपनिषद पढ़ती हैं, उसको जीती हैं, इनके लिए जोरदार करतध्वनि करेंगे। सुंदर प्रस्तुति। अब आज के कार्यक्रम की पूर्णता की ओर हम चल रहे हैं। 2-3 कार्यक्रम और शेष बचे हैं 5-5 मिनट के लिए। तो आपके धैर्य को मैं पुनः प्रणाम करता हूं और आपके बीच में आमंत्रित करता हूं हमारे पतंजलि आयुर्वेद महाविद्यालय की बहनों को सामूहिक सांस्कृतिक नृत्य के लिए। एक बार जोरदार करतल ध्वनि हमारी बहनों के लिए करेंगे। जोरदार अभिनंदन इन बहनों के लिए। परम पूज्य श्री स्वामी जी महाराज, परम पूज्य आचार्य श्री जी की प्रेरणा से ये विद्यार्थी अहर्निश अपने अध्ययन में पुरुषार्थ भी करते हैं और जो थोड़ा समय बचता है उनमें ये अलग अलग प्रकार की कलाओं का भी अभ्यास करते हैं। मैं आमंत्रित करता हूँ पतंजलि संन्यास आश्रम के हमारे ब्रह्मचारियों को जो एक गीत की प्रस्तुति देंगे आपके कर्तव्य ध्वनि के बीच में।
ओम। पूज्य स्वामी हनुमान देव जी द्वारा रचित यह गुरु वंदना आप सभी के समक्ष प्रस्तुत है। गुरुवर मेरा ये जीवन तेरे रास्ते चलेगा, गुरुवर मेरा ये जीवन तेरे रास्ते चलेगा। सुख में हो चाहे दुख में, सुख में हो चाहे दुख में, तेरे वास्ते चलेगा, तेरे रास्ते चलेगा। गुरुवर मेरा ये जीवन तेरे रास्ते चलेगा। तूने सिखाया हमको ऋषि पथ पे आगे बढ़ना, तूने सिखाया हमको ऋषि पथ पे आगे बढ़ना। जाना कृपा से तेरी मंजिल की सीढ़ी चढ़ना, जाना कृपा से तेरी मंजिल की सीढ़ी चढ़ना। संकल्प ये हमारा, संकल्प ये हमारा, अब ना कभी टलेगा, तेरे रास्ते चलेगा। गुरुवर मेरा ये जीवन तेरे रास्ते चलेगा। जीवन हमारा बगिया, तुम हो हमारे माली। जीवन हमारा बगिया, तुम हो हमारे माली। गुरुवर तुम्हारे दर के हम तो है बस सवारी। गुरुवर तुम्हारे दर के हम तो है बस सवारी। मन रूपी पुष्प गुरुवर, मन रूपी पुष्प गुरुवर, तेरे चरणों में खिलेगा, तेरे रास्ते चलेगा। गुरुवर मेरा ये जीवन तेरे रास्ते चलेगा। उद्धार करने जग का योगर्षि आप आए। उद्धार करने जग का योगर्षि आप आए। प्रेमी तुम्हारे गुरुवर हैं आस ये लगाए। प्रेमी तुम्हारे गुरुवर है आस ये लगाए। तेरा भक्त अब ये गुरुवर, तेरा भक्त अब ये गुरुवर, तेरी शरण में पलेगा, तेरे रास्ते चलेगा। गुरुवर मेरा ये जीवन तेरे रास्ते चलेगा। सुख में हो चाहे दुख में, सुख में हो चाहे दुख में, तेरे रास्ते चलेगा, गुरुवर मेरा ये जीवन तेरे रास्ते चलेगा, तेरे रास्ते चलेगा, तेरे रास्ते चलेगा।
अत्यंत ही सुंदर प्रस्तुति। और अब आज के इस सत्र की अंतिम प्रस्तुति लेकर के आ रही हैं पतंजलि विश्वविद्यालय की छात्राएं, जो है आर्टिस्टिक योगासन। आर्टिस्टिक योगासन योगासन का वह स्वरूप है जिसमें संगीत के साथ योगासनों का अभ्यास किया जाता है। और मैं यहाँ उल्लेख करना चाहूँगी कि योगासनों को करने की इन विभिन्न पद्धतियों का जो खोज है वो परम पूज्य स्वामी जी महाराज व परम श्रद्धेय आचार्य श्री महाराज के निर्देशन में हुई है। और परम पूज्य स्वामी जी व परम श्रद्धेय आचार्य जी के निर्देशन में योग आसनों को खेल के रूप में मान्यता दिलाने का कार्य भी किया गया है, जिसके तहत भारत सरकार के मिनिस्ट्री ऑफ स्पोर्ट्स एंड यूथ अफेयर्स ने योगासन को अधिकृत खेल के रूप में मान्यता दी है। और इसके तहत नेशनल योगासना स्पोर्ट्स फेडरेशन व वर्ल्ड योगासना स्पोर्ट्स फेडरेशन का गठन भी किया गया है। और यह अत्यंत गौरव का विषय है कि पतंजलि विश्वविद्यालय के छात्र छात्राएं राष्ट्रीय व अंतर्राष्ट्रीय स्तर पर आयोजित होने वाली इन योगासन स्पर्धाओं में भाग ले चुके हैं व विश्वविद्यालय के लिए, देश के लिए, अपने राज्य के लिए मेडल्स, स्वर्ण पदक, रजत पदक, कांस्य पदक आदि प्राप्त कर चुके हैं। तो अब आपके सामने आ रही हैं पतंजलि विश्वविद्यालय की छात्राएं आर्टिस्टिक योगासन डेमो की प्रस्तुति लेकर। Music, please stop. Please stop the music. Please start it again from the starting.
रघुपति राघव राजा राम, पतित पावन सीताराम।
प्रभु अब धीरज मत देना, प्रभु अब धीरज मत देना। सहते जाए सहते जाए सहते जाए, ऐसा बल भी मत देना, ऐसा बल भी मत देना। उठ कर करने हैं कुछ काम, रघुपति राघव राजा, रघुपति राघव राजा राम। उठ कर करने हैं कुछ काम, रघुपति राघव राजा राम। उठ कर करने हैं कुछ काम।
तुम करुणा के सागर, तुम पालन करता, पर जो जुल्म है करता वो कहाँ तुमसे डरता। तुम पूर्ण परमात्मा, तुम अंतर्यामी, पर फिर कैसे होती रहती हर पल ये मनमानी।
रहा है अब विश्वास। 5 वर्ष धुंधला आकाश, कौन द्वार हम खटकाएं, कौन से वादे दोहराएं। आम आदमी कितना, रघुपति राघव राजा।
हाँ, घर से निकले हैं सुबह, तन मन धन सब है तैयार, खाली हाथ न लौटेंगे, तय करके आए सुबह। अंदर एक सिपाही है जिसने ली अंगड़ाई है, अब तक सोया सोया था, सपनों में ही खोया था। बहुत हो गया है विश्राम, रघुपति राघव राजा राम, रघुपति राघव राजा राम, रघुपति राघव राजा राम।
बहुत सुंदर प्रस्तुति। मैं हमारे बच्चों के चेहरे पर जो उमंग, उत्साह है, जो हर्ष है, उसे देखकर जो आप लोगों के मुखमंडल में जो हर्ष और उत्साह दिख रहा है, तो ये ज़रूर मुझे यकीन है कि आप लोगों ने इस कल्चरल प्रोग्राम को, इस पतंजलि के कल्चरल प्रोग्राम की एक झलक को आप लोगों ने बहुत अच्छे से एंजॉय किया। मैं एक बार आप लोगों से जोरदार करतल ध्वनि की प्रार्थना करती हूँ ताकि हमारे बच्चों का उत्साह और बढ़े। बहुत कम समय में, जस्ट 1-2 दिन के अंदर इन्होंने इस प्रोग्राम को तैयार किया और मुझे लगता है सुबह से 9 बजे से जैसे आप बैठे हैं, आपकी थकान जरूर दूर हो गई होगी। और इसके साथ साथ मैं इन बच्चों के टीचर्स के लिए भी एक क्लैप चाहूंगी, क्योंकि जिन टीचर्स ने इनको सिखाया है, जिनके निर्देशन में इन्होंने तैयार किया है, वो भी इसके लिए बधाई के पात्र हैं। मैं पुनः धन्यवाद करना चाहूंगी प्रोफेसर महावीर अग्रवाल जी का, आदरणीय प्रो वीसी सर, जिन्होंने बहुत कम समय में ही, निवेदन मात्र से ही इस पूरे कार्यक्रम की रूपरेखा को तुरंत तैयार करवा कर आज आपके सामने इस रूप में प्रस्तुत किया। बहुत बहुत धन्यवाद सर। और इसके साथ मैं ऑर्गेनाइजर्स का और उसके अलावा पतंजलि यूनिवर्सिटी के इस भव्य प्रांगण में जितने भी हमारे जो लोग लगे, जितने भी कर्म योगी भाई बहन लगे, हर कार्य के पुरुषार्थ में लगे, एक इस इवेंट को सक्सेसफुल बनाया, मैं उन सब का हार्दिक धन्यवाद करती हूँ। इसके साथ साथ मैं आपको कल के लिए, जो कि कल का कार्यक्रम इस पूरी इंटरनेशनल कॉन्फ्रेंस का प्राण है, तो मैं आप लोगों से अपेक्षा करती हूँ कि कल के कार्यक्रम में भी आप इसी उमंग व उत्साह से शरीक होंगे। सुबह 9 बजे से ही वह कार्यक्रम शुरू हो जाएगा और मैं अपेक्षा करती हूँ कि 9 बजे ही, जैसे आज थोड़ा आने में विलंब हो गया था, तो मैं पुनः निवेदन करती हूँ हमारे यूनिवर्सिटी ऑफ पतंजलि के स्टाफ मेंबर्स जो डिसिप्लिन कमेटी में हैं, पतंजलि आयुर्वेद कॉलेज के स्टाफ मेंबर्स जो डिसिप्लिन देख रहे हैं, कि कृपया करके महाराज जी की उपस्थिति से पहले ही सभी बालकों को यथा योग्य स्थान पर बैठाने की कृपा करें। बहुत बहुत धन्यवाद, बहुत बहुत धन्यवाद आप सबका।